Distinguished guests, ladies and gentlemen, good morning and welcome to International Conference on the 20th anniversary of Bridging Study Evaluation, Ethnic Factors in the Acceptability of Foreign Clinical Data in Global Regulatory Convergence. This event is co-organized by Center for Drug Evaluation and the TFDA. First of all, now please welcome Dr. Xiumei Wu, Director General of TFDA and Chairperson of Center for Drug Evaluation, to deliver her welcome remarks. Welcome, Dr. Wu.
Our distinguished speakers and guests, ladies and gentlemen, good morning. On behalf of Taiwan FDA and the CDE, it's my great honor to welcome you to attend this international conference. Actually, it is a very important conference because we try to reduce and prevent the unnecessary clinical trials. So we have to use bridging study evaluation and to use the global data, all the foreign data. Those clinical trials have been implemented very, very seriously. So I think after our evaluation, if this one can be used, we have to use them. That's why since 2000, we try to use those foreign clinical data and also hope can use them in the NDA. So right now, we want to celebrate these 2 decades, what we have done, and how we to use them, and in the near future, what we should do. So today, there are many topics. I think our speakers will share their experiences, and also hope we can get a lot from you. Today, we also have foreign guests. They are experts, and they have their own expertise in this field. So we can learn a lot from you. So I want to thank you so much. As I know till now, there are more than 600 online audience. So thank you so much. Today, it will be a wonderful day. And I hope our conference will be successful. So finally, I also want to thank the colleagues in CDE. My colleagues, they work so hard. During this pandemic, we do day and night, 24 hours. And I hope we can do something for our country and try to help and also to do something in this pandemic. And I think we have done it. And we also do many things, not only in vaccines, but also in pharmaceutics. So right now, I want to thank you again and hope we can cooperate and we can have more success in the society to help people and also to do something for the patients. So I wish we have a successful conference. Thank you. Thank you so much.
Thank you, Director General. Thank you very much. Thank you. Ladies and gentlemen, our next guest of honor is Professor Oliver Youpu Hu. Professor Hu has made tremendous contribution to bridging study evaluation by putting policies in place when he was Director General of Bureau of Pharmaceutical Affairs in year 2000. Please welcome Professor Hu onto the stage and say a few words to us. Welcome.
Thank you. You'll take over. I should call commissioner instead of a director general. I was the director general. You become commissioner. Commissioner and CEO of our CDE, and also so many old good friends. I don't have time to call you, each one of you, because I only have 5 to 10 minutes to the most. But I review last 30 years data from literature. I will share you just a couple of slides and also some of my new thoughts to share with you. This is the one. I was Director General in charge Bureau of Pharmaceutical Affairs for 2 centuries. What are you talking about for 2 centuries? It's from 1998 to year of 2002. So it's really across 2 centuries. And there's something happened within that 2 centuries. We, together with the Bureau of Pharmaceutical Affairs colleague, together did some little things. For example, since Ms. Xiao have already established several good foundations for our clinical trial. You see, we at that time in 1998, we have already have a relative sound clinical trial conduct in our country. Although people still say a lot of need to be improvement. But after that, we start to implement our GCP seriously. And also we can use this clinical trial, look into the PK/PD of the drug, which may want to apply NDA in our country. And also look into the pharmacogenetics about the drug. So based on this, also efficacy and safety have already approved in other countries. So the new chemical entity or new drug try to launch on our market, they also need to look into anything related to ethnic difference. That includes intrinsic ethnic factors such as genetic and so on and so forth. And also, more important, is extrinsic ethnic factors, including environmental, including this and that, diet, and also medical practice. The different medical practice will cause different results. So that was the consideration of ethnic factor at 1998. So there's when you see that figure, the previous figure, if you are a drug developer, you will think, "Wow, my goodness, so many studies need to be done before we can get licensed." Excuse me. Just like the troubled water across in front of us. So I thought maybe we need something to bridge over the troubled water. This song is the number one song for all time, published in 1970. So if you still can sing "Like a bridge over troubled water, I will ease your mind." That's the last sentence of this song. Okay, so we start our bridging study, bridge that troubled water. Before we start our bridging study, we always follow some regulations, law in the world, have already published in the world. The first one is published in 1998 February, which called ICH E5. There's only one important thing, Commissioner Wu have already told every one of you. We try to minimize duplicate clinical data and facilitate acceptance of foreign clinical data in a new region, for example, in Taiwan, Republic of China, on safety, efficacy, and dose regimen. The reason why I put dosage and dose regimen, there are reasons I will show you later, very quickly show you later. And the guidance will permit adequacy evaluation, and the guidance should implement relating genetic and extrinsic. Okay, so this is the most important slides, but may not exactly the same as the one you will see in the following from the foreign speaker. There's only one reason, because some of us made history, and some of us read history. That makes some difference. Okay, you see, our Vice Minister, Dr. Zhang, mentioned what happened since year of 2000. You see, year of 1993. Oh my goodness, 1993. Here, 1993, July, 777 announcement. This is something related to our Vice Minister Zhang. Am I right? Chairman Zhang. Okay, this is a very important one. I was in Johns Hopkins at that time. See, there are 6 announcements or 6 regulations related to the ethnic differences related to bridging study evaluation since 1999 July. Division head, July 1999. All this I have copy. I have evidence copy in my hand. Even I bring today with me. So the first announcement is for BSE implementation in Taiwan was 1999 July. And then moved to next century, year 2000 March, the second announcement for BSE implementation in Taiwan. And the third one is the 12 announcement, 12, 7. You see how interesting. We always like double, 7. although it's 777, but you know what happened in 777? The Japanese invade China. So we select a good day, 12, 双十二公告, to replace 777. Of course, there's something different. Instead of a 40 or more subject needed, Taiwanese subject needed for apply Taiwan license. Now we try to replace this 40 people's data, use bridging study. Okay, after this, then December of that was let me see, that was this one. The angle is different. Okay, and then 1 year later, about this year 2001, December, checklist announcement for BSc. This checklist is very important because just like Commissioner Wu just said, this conference is important. After we announced this checklist, within 6 months, Thailand exactly follow our checklist and made an announcement. And also personally, Director General of Thai in charge Drug Bureau told me, do you allow us to copy? I said, sure, go ahead, copy. Because Thai and Taiwan are so close. Our TFDA, the same as Thailand TFDA. So once I suggest to the Director General of Thai FDA, we can change our name, CDE, into JCDE, Joint CDE, for Thai FDA and for Taiwan FDA. So once we approve one drug, then there are about 5 times people will use the drug than Taiwan. Because, oh, not 5 times, maybe 4 times or 3 times. Okay, let me quickly to the last. So the last one will be, this is the implementing schedule. And then in the year of 2002, May, the guidance for BSc announced. So this 6th announcement is within these 3 years, relates to today's topic. Okay, after E5, the ICH announced another one, E17, mainly dominated by Japanese government. It's a very, very good one and a very important one, which I think is around November. And this one just move from countries bridging study to multiple region. So data from multiple region clinical trial can be accepted by regulatory authorities. More important, it should be used together with other ICH guidelines, including E5. That means if a country like Taiwan, without joining the multiple region clinical study, they still can ask for bridging study conduct in Taiwan. It says very clearly in the literature and also in the ICH announcement, says it should be used together with E5. Okay. There are many publications since 1991 all the way to 2021. I reviewed so many of them and then just pick up several important ones to show you. That includes many patients. We all know the ethnic or racial differences in PK/PD has been attributed to the distinctions in the genetic medical practice and so on and so forth. And also affected by socio-economic background. This is especially for the pain treatment and cultural diets and environment. All the references I listed over there. Magani did a huge job. He synthesized, this is not 合成, synthesized 综合. He synthesized 20 years of cumulative evidences in the United States for racial, ethnic disparities in the analgesic treatment. What they found? They found that for the subgroup of minorities, the size of the difference was sufficiently large to raise not only normative, but quality and safety concern. That means, you see, for this, use this cumulative evidence for this analgesic treatment, pain treatment, not only the efficacy, but also the safety concern. That was significant. And this is the biggest one. The Brewster and his associate, after retrieve 4,596 reports, and year 35 trial with 7 classes of antihypertensive drugs. In how many patients? In 25,540 African patients. And they found, and the trial with mobility and mortality. This is important. Not just PK/PD, it's mobility and mortality included. And they found
using almost standard treatment for antihypertension patient associated with a greater instance of stroke and sudden death in the Africa patient. This is a solid evidence to show there are ethnic differences. And the same study, the same review from the same group of people from 1581 reports. And how many patients? About 1,719 South Asian patients. But this part of the study without morbidity and mortality data, but shows no difference, no evidence of difference efficacy of antihypertensive drug in South Asia. So this is a need. There is a need for trials with morbidity and mortality outcome to show really they are the same or not. Okay, and also in United States and European country, there are something different. Even in the label, they are different. Taxonomy, the classification is different. I will not go into detail because this is the time limited. A little bit more thought I will contribute to you and share with you. Besides the morbidity and mortality, genetic polymorphism, this term is much better than interethnic. So intergenetic better than interethnic. I believe you all agree with me. Second, non-genetic factors, that's extrinsic factors such as diet, weight, medical practice, and the environmental factors, especially the weight. You see, usually we give a dose for one person. A person with 40 kilo and with another person with 140 kilo. My goodness, you give the same dose to them? No, you don't. But this is also some ethnic differences come from. And also, this is should be highlighted as potential sources of inter-individual variation in PK and PD of the drug. So please, don't neglect this part of the importance when you apply your new regulation. There's another one we almost ignore, which is the use of different dosage form, dosage regimen, specific administration route may trigger significant differences PK/PD response in various ethnic groups. These have largely been ignored or overlooked. Not just I say this, our good friends Chen Meilin. Now we have another troubled water in front of us now. The three thoughts I just shared with you, and also different dosage regimen, different dosage administration route may cause ethnic difference. And also morbidity, mortality is the final outcomes we can. So, like the bridge over troubled water again, we will together solve the problem. In the long history of fighting with disease, thousands of years, this 20 years is just a very good start. It's just a very good start in this long history. Much achieved, we together did, but more to do. Thank you very much.
Thank you so much, Professor Hu. Thank you. Please return to your seat. Thank you. The bridging study evaluation of new drug applications according to ICH E5 guideline has been implemented more than 20 years in East Asian region. In the past 2 decades, the implementation of regulation and practical operations of BSE has critically facilitated the process of new drug development, review, and approved by TFDA. The adoption of BSE requirements apparently resulted in an effective regulatory pathway for extrapolating foreign clinical data to demonstrate population and further in minimization of unnecessary duplicated clinical trial, just like Director General just mentioned. And now we're going to, it's really an honor that we have the presence of so many distinguished guests today. And now please may I invite the following guests of honor onto the stage for group photos. 那我们接下来欢迎接下来倡导名的贵宾上台，一起跟我们合影留念。Please welcome Director General Dr. 秀美武。Please welcome Professor Oliver Youhu Hu, Chief Executive Officer Mr. Kevin Mingxun Liu, Chairman Hong Ren Zhang, Professor Chun Xiu Gao, Professor Li Hengbao, and Professor Jing Fu Xiao. Dr. Du Xianjian, Deputy Executive Director Li Juanxu, Deputy Executive Officer Shou Yulin, Dr. Heng Dechen, Dr. Mei Wang, Section Chief Ting Yazhang, Dr. Yizhen Sun, Dr. Ming Xiaozhan, Dr. Jiaxin Ye, Dr. Jixun Chen, and Senior Reviewer Ms. Yiling Wang, and Project Manager Mr. Jiren Yang. And there are also 3 distinguished guests who will take group photos with us online. Dr. Junko Sato from PMDA Japan, and Ms. Louise Watt from Novartis Singapore, and Professor Wenhua Guo. Okay, now please look at the cameras in front of you. And for our online guests of honor, please look at your webcam. 3, 2, 1. Okay, please give us a thumb up. 请把大拇指借给我，我们一起预祝今天的研讨会顺利成功. Together, we wish every success of this conference. Thank you very much. Thank you. Please give a big round of applause to all our distinguished guests. Thank you. And we thank our online guests of honor. Thank you for joining us for group photos. Thank you. Thank you. Thank you very much, our guests of honor. Thank you very much for your generous support to this event. This conference aims to review the status and execution of ICH E5 guideline and to share perspectives from the regulatory authority, academia, and the pharmaceutical industry. Now, ladies and gentlemen, we will proceed to the first session. We are very honored to have Professor Oliver Yopu Hu again as our moderator. Please welcome Professor Hu onto the stage.
Before I introduce Dr. Chen, I will add several things. First one, for the last 22 years, CDE, all the members of CDE, did a lot of great job, especially today's bridging study. I have been thank you for all the effort you have already put in. And also our previous Bureau of Pharmaceutical Affairs colleagues, they also not 24 hours a day, 25 hours a day, 8 days a week, work for not only bridging study, but also all kinds of things our people, our patient interest. And Dr. Chen obviously is one of the leader, actually is an important leader, not only for this bridging study, for many things happen for the last 20-some years. Dr. Chen had his MD degree from National Taiwan University many, many years ago, because I heard I think it's around April, I heard Dr. Chen going to retire next month, 31st, am I right? So this month, May, this month. May have 2 meanings. One is May, May retire. Dr. Chen had his MD degree and then later on get a PhD degree from, at that time, the most famous liver transplant university, which is University of Pittsburgh. His PhD degree relates to clinical pharmacology, and also he was one of the pioneers in Taiwan, knowing not only the medical practice, but also pharmacokinetics and pharmacodynamics. And he very early he implemented many regulations in National Taiwan University, such as to visit their the study conducted, the clinical study conducted in National Taiwan University. And in 1998, we recruit famous Dr. Chen from National Taiwan University to our newly founded CDE. I think I remember Dr. Chen arrived earlier than the CEO to our CDE. At that time, we start from 14 people, from 14. Now almost reached to 400, I think, or more than 400 now? 300, very good, very good. So he spent almost all his precious career time in CDE. Not only start the institute, but also make the institute prosperous. And until he retired from CDE, CEO, and still contribute in the CDE for some time. Then he moved to academic Seneca incubation center, which is the window of our country for biotech pharmaceutical industry. He was the CEO for some time. And we always work together. when you were at Academic Seneca, I am in Academic Seneca. So until today, he moved back to CDE, now become a consultant. And I don't think people will allow you to retire. So no wonder you select May to retire. He's going to talk about 20th anniversary. He's going to talk about 20th anniversary of ICH E5 bridging study, reflection and a future perspective. Let's welcome Dr. Chen.
Thank you for the kind introduction. Today I will talk about the 20th anniversary of ICH E5 bridging study. Reflection, I will remind the history in the past and present, and then look upon the future perspective. So because this is a topic most relevant to the East Asia and beyond. So I will first talk about why we implement ICH E5 and how we implement it, and what's the content, the procedure, then what it brings, the impact, then talking about the future perspective. So in 1995, I went to the Yokohama to attend ICH3. Immediately, Taiwan recognized this is a very important global regulatory harmonization movement. So we start a project called ICH in Taiwan since then for almost 10 years. And then we attend ICH4, ICH5. And I quickly catch the importance of E5 announced in 1998, because this is the guidance most relevant to Asia in accepting foreign data. And as Dr. Oliver Hu just mentioned, I was recruited to the CD around that time, 1998. And also Dr. Hu was become the Director General also in 1998. And both of us served in the advisory committee for Ministry of Health for many years for new drug evaluation. And I reviewed the clinical part, and Dr. Oliver Fu reviewed the preclinical part. So with both of us, we catch this important guidance. And also I have my PhD in clinical pharmacology, actually major in pharmacogenetics. So we both know this important guidance. And at that time, we have a 40 Taiwanese trial subject requirement for NDA registration since 1993. And this is an important announcement to set up the clinical trial infrastructure, but we always feel also receive some criticism from global companies that this is like, we call it registration trial, because the 40 case is not based on statistic analysis, and the protocol design is not particularly try to answer any ethnic sensitivity question. So of course we try to phase out this announcement, and then ICH E5 becomes a good chance to phase in, to replace it. And we also find this probably what East Asia can contribute to the global regulatory science, because this is something we very much emphasize. So we can have a strategy, have a voice to engage ICH and through many related international platforms like APEC, like DIA. And at the very beginning of CDE, we have support from US FDA to send Taiwanese reviewer back to Taiwan to help us. So I learned from them what to do good review practice. And I define it as qualified regulatory reviewers, adherence to pre-specified standard in terms of content and process. But at the very beginning, we have very limited resources in staff, personnel. So every global company looking upon this emerging market, their first question, can you do good review? And do you have good regulatory science concept, just like EMA, FDA? And also, you are such a small market. Taiwan only has 0.68% of global pharmaceutical market. So you absolutely have no bargain power to talk to them what you need for registration. So better no further question asked. Otherwise, you might have to wait because we don't want to do it just for your request. And that limitation turned out to be our strength, because we know we don't have any bargain power unless we base on best good regulatory practice, then we have a chance to talk to those global companies. So try to develop the GRP. Not only we have full-time staff, and then our review is not only based on the content, but also you have to have some guidance, some template, and have to be evidence-based and also risk-benefit evaluation and consistent, and you have to have quality control. That's done from FDA. So the process has to be transparent, predictable, timely. So we set up project manager system. And Taiwan, after so many years, we now chair a project called GRP Center of Excellence under APEC. So when I look at E5, I found they have some merit, such as they define the concept intrinsic and intrinsic factor, ethnic sensitivity, BSE, pathway, Q&A. But they leave the detailed process and requirement to local regulatory health authority. So the cons is that this vague concept, sometimes regulatory authority will say, oh, okay, we deal with it in a case-by-case scenario, and they don't develop a systemic approach and no clear regulatory pathway. So different authority might potentially disharmonize because they have different interpretation. And the spirit of ICF E5 is try to avoid duplication of clinical trial might be lost. So when Taiwan start to announce we want to do BSE regulatory pathway, immediately drew global attention of industry. They suddenly, everyone come to knock my door, say, how are you going to do it? Because they are afraid we might go on the wrong direction and others might follow. Because at that time, there's no model of how to implement ICH E5 at that time. And certainly, many regulatory authorities also like to know how you are going to do it in Asia, because this is most relevant to East Asia. So we see this table again and again, but I try to integrate the concept into BSE assessment aid and form a regulatory pathway that is operable. So first, I try to have a working definition of ethnic group, because if we define ethnicity as a race, there are a lot of exceptions, because race didn't catch the extrinsic factor. Or citizenship. In a country, you might have many different ethnic groups. For example, there are 6% Asian in the United States, 14% of Black American in the United States. So you have multiple ethnic groups. And geographical region also have a lot of exceptions. But what we deal with in NDA is the patient taking the drug. So we should only focus on the drug characteristic of this particular drug, what is pharmacogenomic in ADME, PKPD, and also we have to focus on intra-ethnic variation, not only inter-ethnic variation. And is the dose response curve very steep or therapeutic window very narrow? Any drug-drug interaction with the current practice? And the patient I define as epidemiology of indication. This particular target population, for example, lung cancer, we are not dealing with all the lung cancer. Some indication only go to non-small cell lung cancer, like gefitinib. We feel there is an ethnic difference because EGFR receptor mutation is different. So you have to go into the incidence etiology, nature's pronounced previous response to related medication, medical practice, or resistance for virus or bacteria, local clinical experience. That we feel that Equinox, this particular case, Asian had more headache, and Dr. Chen Jixuan will talk about that example later, potential for abuse. So we put it into a template and divide it into preclinical and clinical, 12 questions. And then at the end, we say we have to evaluate in totality based on clinical impact and risk management, because you don't want to delay the marketing of this product, asking for more data because patient is waiting. If we can manage the risk post-marketing, then we do that too. So I simply put it into a slogan, DIC, drug, characteristic, indication, clinical. And this is exactly the same slide I present in ICH 5, Global Cooperation Group Pre-Conference, San Diego, year 2000. Because I keep saying that everyone feel very interesting, so they invite me to have this talk. One key question is that, can we accept East Asian data, or we need Taiwanese? Because sometimes people say, yes, if you ask 40K Taiwanese, then China, Korea, Japan, they all ask their own data. But we say we accept all East Asian data. Because at that time, I found an article published by Professor Lin in Taiwan. She used the HLA typing to differentiate the genetic distance among all the ethnic groups in Asia. And she came to a conclusion that Asian can barely differentiate into Southern Asian and Northern Asian, and divided by the Yangtze River. So you can say Japan, Korea, Mongolia, Northern Chinese, they belong to the Northern Asian. And then many Southeast Asia or Southern Taiwan, Southern Chinese belong to this group. This is Southern Asia. But I noticed that in 1949, there are 16% of Taiwanese moving from China to Taiwan. And in our clinical observation, we've almost found no difference. So we feel that Asian and East Asian, we can accept all their data as a group. And then you will talk about, how about Asian data in the United States? It's a little bit different because the extrinsic factor may be different. We sometimes call this group as a chopstick region because we use chopstick. So the extrinsic factor is much more similar. So BSE become an evolving regulatory policy. And how we want to implement it, we will try to harmonize with E5, have clear pathway, and we want to communicate with all stakeholders through education, consultation, a lot of publication, international platform. And we also offer a transition period. You can choose to do 40Ks or you want to do BSE. And we also have 5 announcements. There are some category very unlikely to have ethnic sensitivity. We say you don't have to do it. And then we publish our result. Like the first 3 years, we assure everyone that almost 80% are waived for further bridging study. Even you don't have any Asian data, the rate of waiver is about the same. So companies suddenly change from against it, become to embrace it. They say, okay, this is a good result. Perhaps other countries can take a look at Taiwan's experience. So this is our checking list. we put it into an assessment aid that asks company to do self-evaluation. This is this in 2003. So you can see we asked the company to put this almost like a bridging data package in the complete clinical data package. Where is your data? Now, certainly you can hyperlink with electronic data. And then this is the drug characteristic. We have question 1 to 8. And then 9 to 12 is clinical question. So company can do their self-evaluation. And this is a very advanced concept in regulatory science, because not until 2019, US FDA started to have this assessment aid for self-evaluation in their OCE, Oncology Center of Excellence. And we do it 2003. But at the end, the spirit said you have to have all data in totality to evaluate their risk-benefit. And after company do their evaluation, our CD staff would do our evaluation. So this is one of the slides. You can see we published a lot of paper in Chinese, in English, and have conference poster and presentation in everywhere. So this is one of the DI. You see, we have 7 CD staff in that meeting. Everyone have a poster or podium presentation, and also we have DI Taiwan night. We make a lot of friends, have a lot of fun, and let people know what we are doing. So the background is how Taiwan is going to accept foreign clinical data, and why you do clinical trial in Taiwan. So what's the impact? When we announced BSE, suddenly the composition of clinical trial in Taiwan have a dramatic change. This blue line is you do clinical trial together with all international. It's almost like an MRCT trial. And this is the registration trial just for Taiwan. Suddenly, 90% of our trial become international trial. So you can see at this time, from 2000 to 2006, Taiwan had the most experience, become the most experienced country in Asia to do MRCT. And the trend keep up to now. Now we have over 380 trials a year. And we also say we are a small market. How about we put it into a bigger market like APEC? It composes 65% of global market. So we go to APEC and we host a project called Joint Research Project on Bridging Study for 8 years. And Japan, Korea, Taiwan, we have a lot of discussion. But the purpose is try to partnership in harmonization with ICH. So since I give so many talks, Thai FDA director, or nickname called me Mr. Bridging Study. You come again. So also because of this publication, one day I get information from an MIT PhD student, Dr. Wen Hua Guo. He will give a talk later. He said he's doing a PhD related to the history of science with impact. And he suddenly find Taiwan has someone talking about bridging study in an international platform. So he wanted to choose this as his dissertation thesis. And he visited me and discussed with me about 100 hours. And today he became a professor and he would give his talk. And in 2009, he published The Voice on the Bridge. So he will give a talk later. And I become invited speaker to many occasions, including ICH 5. And because we are our APEC study, we accepted into the Global Cooperation Group and later ICH. So today is a good time because ethnic sensitivity is still a hot global topic. In March, FDA rejects one BLA from Eli Lilly because they use a single phase 3 trial in China, try to apply and BLA. And FDA said they didn't consult with FDA. Their protocol design didn't represent the population in the United States because the racial ethnic diversity. And 4 days ago, another project similar rejected by FDA because there are 25 similar projects. So FDA even published in April, FDA issued a draft guidance for increasing racial and ethnic diversity in clinical trial. As I said, 6% Asian American, 14% Black American never get representation in clinical trial enough. So FDA started to look at this topic. So MRCT is a very good, but BSc can be a self-assessment aid for early development and Asian inclusive MRCT planning. Why they don't come to FDA for consultation? They don't have tool, but this is similar to assessment aid. I think many regulatory agents should develop. And by early involvement, you can build in all the potential ethnic factor into MRCT protocol with good sample size and pre-specified statistical analysis. Otherwise, you go back to data, try to have indirect comparison. Many data are not collected, and the comparison is not fair enough because it's not done in parallel. But in rare case, if this evaluation finds some ethnic sensitivity question, then you should design it even into a separate protocol, but do it together. And I think with all those MRCT well designed, the BSE will be minimized, but not completely replaced. Future perspective, I think the format of BSE assessment aid should become a tool, and I wish more regulatory agencies can use it for consultation. And this should become a separate section in review report, even put it in public. CDE might try to do that in the future. And we should have regulatory dialogue and convergence, especially in many platforms like FDA Project Office, and I think Dr. Junko Sato will also talk about their initiative about Asian regulatory harmonization. And I think the major findings should also put in a drug labeling and updated by post-marketing data. So thank you for your attention.
Thank you. Thank you very much, Dr. Chen. Thank you. Now, may we please invite Dr. Hu onto the stage again to introduce our next speaker for us, please.
The conference didn't arrange discussion time or question answer time, am I right? So since Dr. Chen going to retire May 31, so you have plenty time to raise any question to him in any place. Or you have anyone have any important question to raise? Just one question? If there's not, Dr. Chen gave a very conclusive opinion and history about ethnic differences, but not only ethnic difference issue, but also good regulatory review, and all the way to actually imply how Taiwan established our clinical studies foundation. That was very important, and I appreciate very much. Next speaker will be the section chief, Zhang Tinya. Oh, good. I cannot find you. Zhang Tinya, he graduated from Taipei Medical University School of Pharmacy, had a master degree from Medical University, Taipei Medical University, and also she had a bachelor degree from the same university. She joined Food and Drug Administration, I think 2014. And before that, she was the clinical pharmacist in Xinguang Hospital. She's going to tell us more detail about today's topic. It's policies for ICH E5 and E17 implementation. I believe we will gain a lot of insight from her talk. Section Chief Zhang, please. I carry the microphone with me.
Thank you, Professor Hu. Good morning. I'm Tingya Zhang from Taiwan Food and Drug Administration. Today, I appreciate the opportunity to be here with you to share policies for ICH E5 and E17 implementation in Taiwan. This is the outline of the presentation. First, I will briefly introduce the policies for bridging study evaluation. Then I will share with you about E5 and E17 approaches. Then I'm going to state current status of IND, BSE, and NDA applications. And finally, I will show you the future prospects of clinicals in Taiwan. First, I will introduce the history of policies for bridging study evaluation. Since 1993, the clinical trial data, including Taiwanese, was required. And in 2000, the clinical trial data, including Taiwanese, was replaced by CCDP with Asian data and the bridging study protocol or reports. BSE application started. In 2002, guidance for bridging study evaluation was announced, which followed ICH E5, considering ethnic factors. In 2009, regulations for registration of medicinal products, Article 22-1, bridging study evaluation was required for certain kinds of new drug application. And last year, in 2021, the regulations for registration of medicinal products is revised and added 22-2 article. Before 2021, the BSE regulations, new drugs like new chemical entity or new biologics were required for BSE. And after 2021, new chemical entities and new biologics, which has relevant local clinical trial data, and along with pharmacokinetic data on ethnic groups in East Asia, the BSE could be exempted. And drugs which has designation of treatment of pediatric or rare severe disease or cellular and gene therapy products, BSE could be exempted also. And I will show you the E5 and the E17 approaches in Taiwan and how they were implemented. This is the IND review process. FDA has the IND reviewing process. The sponsor and the hospital or CRO can submit clinical trial protocol parallelly to TFDA and IRB. And it takes about 45 days to review the IND application. And IRB permission will depend on the site requirement. TFDA with assistance with the CDE to form an IMPRO to handle the reviewing application and documentation evaluation. For some cases, if there has some ethnic or ethical issue, we consult the advisory committee. This is the IND review timeline. If the MRCT, which can receive the IND approval within 15 days through fast track, and the other INDs with no significant scientific or ethic issues also can receive IND approval within 45 days. But for the INDs with higher risk, it may be consulted with advisory committee. So the IND approval may need 120 days. And for cell and gene therapy, IND fast-tracked is also applicable. It takes 30 days to receive the IND approval. And for those with no significant technical and safety issue, cell and gene therapy IND, the IND approval will receive within 45 days through standard review. And the same if the cell and gene therapy with higher risk, we also consult with advisory committee. So the approval time may need 150 days. This is the BSE process. If a new drug or a new chemical entity or a new biologics, BSE will be required. But if there is representative local data and East Asian PK data, BSE would be exempted. And if there is no sufficient data or CCDP does not meet the regulatory requirements, then we will, if the CCDP meets regulatory requirements, then we will check the differences of the PK and the PD data between the foreign data and Taiwan. And then to determine the bridge study can be needed or be waived. Here are some CCDP regulatory requirements, such as adequate pharmacokinetics, pharmacodynamics, dose response, efficiency, and safety in foreign regions. And clinical trial must be conducted and designed follow GCP. And there must be adequate and well-controlled. Appropriate endpoints and diagnostic definitions may be used to evaluate the treatment. If CCDP meets the regulatory requirements and the foreign data can appropriately extrapolated, so there is no further study needed. And if authority or the sponsor think there is some problems or some differences of ethnic factors to alter the efficacy or safety of the medicine, then a sponsor needs to generate a limited clinical data like bridge studies. And but if the CCDP doesn't meet regulatories, but the foreign data can appropriate extrapolated, so the sponsor may need additional clinical data, and the clinical can designed to serve as a bridge study. But if the CCDP doesn't meet regulatory requirements and the foreign data cannot be appropriate extrapolated, so the sponsor may create both clinical trials and bridge studies. This is the guidance for BSE to guide the BSE evaluation. And the ethnic factors are defined as intrinsic and extrinsic characterization of the population. Intrinsic factors include genetic and physiologic and pathological conditions, such as gender, age, ADME receptor sensitivity, and genetic polymorphism of the drug metabolism. And intrinsic factors include culture or medical practice or drug compliance. Okay. ICH E5 approach, both foreign clinical data and bridging data in Asian, East Asian, or Taiwan can be considered together. And in these past years, ICH E5 was implanted in Taiwan. It pooled regions and subpopulations data in CCDP, and both intrinsic and extrinsic factors are both considered in ICH E5 approach and ICH E17 approach. We extrapolate the clinical data under ICH E5 and E17. The clinical data was conducted in one region, in each region simultaneously. There are some aspects of ICH E17 guideline, such as pre-consideration of regional variability, subject selection, dosage selection, choice of endpoints, sample size planning, connecting and handling of information, statistical analysis planning, selection of comparators, and concomitant medications. ICH E17 can shorten the drug evaluation time, so we use ICH E17 guideline measure. This is the health insurance drug benefit. If a new drug MRCT, which has one of the A10 countries and a certain portion of Taiwanese subjects, and NDA submitted to US FDA or EMA, it can have 10% increase in drug price incentive and the reimbursement will from the national health insurance. Last year, we conduct GCP inspection in compliance with new drug application. In this new system, we improve the efficiency of new drug reviews and strengthen the link between GCP inspection and NDA. And we promote Taiwan's GCP inspection system to be in line with international standards. GCP's inspection results will be reviewed as part of the evaluation of the new drug registration application. And the GCP inspection will not affect the number of days for the application of NDA. This is the NDA expedited process. We have through reviewing track and through reviewing tracks and priority review, abbreviated review, and standard review. Now I will introduce the current status of IND, BSE, and NDA applications. This is BSE applications in these 3 years. We can see that the BSE can be waived in almost 89% of all the total application. And Data with Taiwan group leads to BSE with the mostly. Reasons for bridging studies needs there is not enough East Asian group data, or there is divisions in East Asian group data, or ethnic difference in safety data. The slide provides IND applications in Taiwan by study phases. We can see that increase in early phase applications in Taiwan. And the slide provides IND applications in Taiwan by local MRCT type. We can also see the increase in MRCT applications in past 17 years. And MRCT takes about 70 to 80%. In the early 2020 COVID-19 outbreak, but Taiwan has well controlled, so the clinical trial like MRCT has increased. These slides provide challenges for conducting clinical trials. Because of COVID, we have challenges about recruitment and the retention of participants, and the completion of trial assessment and the trial visits, and the provision of investigational medicinal products, and changes to monitoring, auditing, and inspections. Because the COVID, many countries take the measures to face the COVID. TFDA also took some measures like telemedicine, verbal informed consent, delivering medicinal products for subjects by delegated study nurse, or the third-party logistics, and remote monitoring. This is the guidance on conduct of clinical trials of medical products during COVID-19 pandemic, and the guideline and the guidance was announced in 2020 and revised in 2021. It includes a return visit of trial participants and administration of investigational drug, and reports of SAE and protocol deviations. Finally, I will show you the future prospects of clinical trials in Taiwan. There are some measures to encourage to conduct MRCT in Taiwan to get sufficient Asian and Taiwanese data and earlier access to innovative therapies. The measures include accelerate IND review efficiency, and strengthen consultation system, and consolidate regulatory and clinical trial environment. This is the future prospects of clinical trials. In the future, we will keep being harmonization with ICH guidance, and cooperate with 36 IRB committees to improve the quality and efficiency for clinical trials review. And for human research environment, TFDA will enforce CRO and CRA training, and expanding the e-platform for clinical trial application. As for GCP inspection, we create GCP inspection database, and to evaluate the remote GCP inspection. Finally, we hope we can establish 3 win situations for manufacturer, public health, and the health authority by the ways of accelerate drug approval, ensure drug safety, efficacy, quality, and accessibility, and harmonization with international regulations. Thank you very much.
Thank you, Section Chief Zhang. Thank you very much for your insightful talk. Thank you. Oh, And let's please once again welcome Professor Hu.
We have a couple minutes left. Anyone have questions from online? 有没有任何人在那个 online 上面有问题？待会有综合讨论时间。哦，那是另外，对，我了解了。没关系。所以待会有综合讨论时间，那么你们要注意要去搜那个 online 上面的问题来综合讨论。好，Okay, now back to English time. Chief Zhang, where's Chief Zhang now? Okay. Chief Zhang gave us a very good, very informative talk, not only from policy point of view, but also review all the current status of our clinical study, especially separate bridging study, separate local clinical study, and the multiple region clinical study. And combined with Dr. Chen, Dr. Chen just mentioned all the framework, all the policy together with Department of Health. Chief Zhang really complete the whole talk, including from the announcement to the implementation by CDE, and then come back to the section of medical product, which is your section of medical product. And then through these two talks, including my little talk, will complete our initial interest. If people don't know what happened from the beginning, how we implement ethnic bridging study, now probably they can understand 80–90% of this bridging study evaluation, how this can help. And also, I really like to have Chief Zhang, when you implement, when you deal with all this bridging study evaluation, please don't forget not the principle, but also the details. Like if any genetic differences published relates to Taiwanese people different from other, not necessarily Caucasian, but also the East part Asia people, and also some medical practice different in this country with other country, and more for pharmacy part. If in Taiwan the dosing regimen changed, for example, 3 times a day, compared with 8 hours, give one dose, those will cause some differences. Maybe the absorption different because of the saturation, or you see administration route. If we change from oral to absorb from mouth, will be very much different. If there's something wrong with the liver, I think Dr. Zhang also realized this. So I just want to emphasize, although we have already changed our regulation for bridging study evaluation, I think it's the third slide of your talk, but still, I think we can find some interesting thing from BSE. That's my suggestion. Any question you want to raise from the floor? Anyway, there will be a discussion time. Thank you very much. Thank you.
Thank you. Thank you, Professor Hu. Thank you very much for chairing this session. Ladies and gentlemen, now we're going to take 20 minutes break. Our next session will begin at 11:10. Thank you. And for those who are joining us online, please feel free to raise your question by clicking the link at the bottom of the YouTube live screen. Thank you. We'll come back. Now we will proceed to the second session in the morning. And it is our great pleasure to have Mr. Kevin Mingxun Liu, Chief Executive Officer of Center for Drug Evaluation, as our moderator. Please join me in welcoming Mr. Liu onto the stage. Welcome.
Thank you. The first topic in this session will be presented by Dr. Junko Sato. Dr. Junko Sato is an office director of Office of the International Program at PMDA. She joined regular agency in 1998. She became a review director of Office of the New Drug in 2004 and moved to Office of Safety in 2009 to develop a new risk care management system through life cycle of drugs. During this period, she visited US FDA as a guest reviewer from 2002 to 2003. From 2012 to 2014, she was dispatched to EMA as the MHLW PMDA liaison. She enhanced collaboration between EMA and the MHLW PMDA and brought a huge success as a liaison. She contributed to some global harmonization activities. For example, ICH, GIOMS. She also contributed many DIA activities. She received DIA Outstanding Service Award in 2010. She led activity of PMDA Asia Training Center and contributes to building stronger bilateral relationship with Asian countries by playing the role as the contact point. Dr. Sato going to share the PMDA's perspective on the implementation of ICH E5. Let's welcome Dr. Junko Sato. Sato-san, please.
Thank you so much. Kind introduction for Dr. Liu. So, thank you for inviting me to this memorial conference. It is my honor to make a presentation in this conference. Today, I would like to share Japanese experience and future perspective of ICH E5. Next, please. So this is a circumstance before implementation of ICH E5. Each pharmaceutical company conducted clinical trials in each region and created data package for each region. Usually, the development was conducted in EU and or US, and the company obtains a regulatory approval in EU or US at first. After the approval, the company considered to develop it in other regions, such as Asia, including Taiwan and Japan. If they decide to develop it in that countries, they plan to conduct new clinical trial in that region, similar to those already conducted in US and or EU. Usually, there was no significant difference in the result obtained in each region. It means similar data package was created in each region. Such style, waste, money, human resources, and above all, the population of patients who participated in clinical trials. We must avoid such waste. Under the circumstance, ICH was started. Next, please. This is the history of ICH E5. As you may know, ICH was established 1990. It means the discussion on ICH E5 was started shortly after ICH was launched. At ICH, what information is needed to support adults for registration was discussed in 1919 first. We Japanese regulator also joined the discussion. Usually, registered dose in Japan is lower than EU and US. The reason were considered scientific factors such as body mass and pharmacogenetic factor, and also non-scientific factor that Japanese patient and physician place a high value on lack of side effects. It also comes from partly complexity of issue difference in medical practice, diagnostic, and ethnic difference in different regions. It was hoped ethnic and/or environmental difference was included as one of the subjects to be discussed in ICH, and classification of effect and the nature of difference would be contributed. The promotion of mutual acceptance of clinical data. Under such discussion, ethnic factor in accessibility of foreign clinical data, ICHE5, was adopted as new topic for ICH. The first expert working group was held in September 1992, Tokyo. Step 2 and 4 achieved in March 1997 and February 1998, respectively. It means it takes 6 years for finalization. In Japan, it was translated to Japanese and published as Japanese regulation in August 1998. After the release of the guideline, 2 Q&As were published in 2004 and 2006, respectively. Next, please. This is the objective of ICHE5. As I mentioned earlier, we have a responsibility to use of any resource effectively. Development cost of pharmaceutical products are paid by pharmaceutical company, but budget is not infinite. Therefore, we all stakeholders has to consider effective use of the budget, human resource, to provide innovative product to patients. To minimize duplication of clinical data, extrapolation of foreign clinical data to new region is very important. So in the process, bridging study is proposed. Next, please. Okay. This is the concept of bridging strategy. In order to utilization the strategy, the existence of the complete data package submitted for approval in other country is required. The right column is the complete clinical data package in other submitted to foreign regulatory agencies. If I explain the strategy very simply, developers will conduct PKPD study and bridging study in new region to compare the result with complete data package. If the similarity is confirmed, result of this study can be extrapolated to data package in new region. It means a developer can skip to conduct this study in new region. The developer can collect necessary data to obtain regulatory approval faster and at low cost. In the other words, developer can spend human resource and development cost to other new product. We are regulator, not a developer, but regulator also always have to consider such things for patient. If regulators require nice to have data and duplicate data with other regions, the number of medicines that can be developed by pharmaceutical company will decrease. However, if the patient's background, medical environment, etc. are different, and it can be extrapolated to Japan. It cannot be extrapolated to Japan. In such case, extrapolating these data to Japan would be a disadvantage. advantageous, and bridging may not always to be best development strategy. Next, please. So I'd like to go back to ICH E5 guideline. Another important idea brought about by ICH E5 is ethnic factors. Ethnic factors are defined in ICH E5 as shown here. The factors are divided to extrinsic and intrinsic. At the beginning of ICH E5 discussion, available pharmacokinetics data were collected and analyzed. As a result of analysis, difference in days is not bigger than individual difference in A days. Based on the result, ethnic factors are classified to intrinsic and extrinsic factors. Next, please. This is the most famous figure of ICH E5. Ethnic factors are divided to intrinsic and extrinsic. Intrinsic ethnic factors are categorized as genetic and physiological, pathological conditions. This concept is so important not only when using bridging strategy, but also when using foreign clinical data. We always consider these factors to review MRCT, etc. We always consider such factors. Next, please. I'd like to touch a specific experience of utilization of ICH E5 in Japan. This is a case of oseltamivir, which was approved using bridging strategy. Clinical trials conducted in Japan are shown the left column. Right column shows the foreign data, complete data package. In Japan, these two clinical trials are conducted. Both Japanese and Caucasian were enrolled to center column of trial to compare PK in both population directly. Based on result of these data, the foreign clinical data were judged to be extrapolatable to the Japanese population. And the trial result in red box were used for review in Japan. Next, please. This is the annual transition of pharmaceutical product approved using the bridging strategy. Blue shows approved products utilize the bridging strategy, and gray is approvals not utilize the strategy. You can see percentage of utilization of bridging strategy is high in early 2000. Recently, the percentage is very low because MRCT is getting measured in Japan. Next, please. So bridging strategy is utilized in many countries, including Japan. It helped to avoid duplication of clinical trial. To utilize bridging strategy, foreign clinical data package is necessary. It means development and approval in other countries is necessary. To avoid drug lag, different development strategy is essential. Next, please. This is a personal opinion, but I think trend of clinical development is shifting as shown here. At first, most clinical development were conducted in Japan as domestic trials. Based on the data, MHLW PMDA gave regulatory approval to industries. After implementation of ICH E5, strategy was often utilized. To utilize bridging strategy, foreign clinical data package is necessary. That means that development and approval in Japan are delayed from other countries. The strategy is so helpful to catch up behind, but it means the existence of drug lag. That is a problem. Not to make new drug lag, another strategy is necessary, so PMD encouraged industry to shift from bridging strategy to MRCT. Therefore, we publish basic principle on global clinical trials in 2018. Sorry, in 2007. And 2018, ICH 17 is implemented. Currently, major trends shift from bridging to mutual development with other regions. Next, please. But I believe ICHE5 has brought us not only bridging strategy, but also useful concept. They are ethnic factors and extrapolation from one population to other population. These concepts are highly applicable in many situations. We frequently use these concepts in our review now. Next, please. So this slide shows the number of clinical trials in Japan. Blue line shows the percentage of MRCT. This percentage is increasing year by year. Red line shows percentage of Asian clinical trials. The percentage is still not so high, but we would like to increase Asian study. As many speakers told so far, so Asian has a very similar factors. For the purpose, we are making effort. I will let you know our perspective to Asian study using a few slides. Next, please. Light graph shows the similarity of COVID-19 mortality in Asia. Last years, we are comparing the mortality in Asia with other regions. Other region before pandemic of Omicron. But I think the Asia as well as COVID-19 has high ethnic. I think we Asia has a high degree of similarity in ethnicity. For example, I'm a Japanese. Most of today's participants are Taiwanese, I think. Perhaps it is impossible to distinguish between Taiwanese and Japanese when you look them. On the right side is the intrinsic and extrinsic factor of ethnicity described in ICG5 as shown. Looking at the individual factors, we have many similarities. In view of this point, I think the Asian data is highly extrapolatable. So next, please. Needless to say, Asia is where we live. Share lots of similarity in genetic point of view. This also means there may lie the difference between Asian population and Western population. So that's between Japanese and Western population. If exposure is different between Asian and Western, and the response is equivalent, we have to choose different dose from Western. How to assess appropriate dosage for Asia? We can share the knowledge and experience with Asian country on review focusing on ethnic factor after implementation of ICHE5 1998. We have lots of experience in this area. Through Asia Training Center, we are sharing our experience. We can also contribute to development and deliver of safer and more effective drug, which means public in Asia through regional cooperation. Because Japan is a country in Asia, it will also be benefit to Japan. We can bring better medicine product, better medical product to patient in Asia together with Taiwan and other Asian countries. In the next few slides, I would like to introduce what the government of Japan is working on to achieve this. Next, please. This slide shows the basic policy for Asian Human Well-being Initiative, which was determined by Headquarters for Healthcare Policy, Government of Japan in 2016. It aims to promote bilateral and regional cooperation that foster sustainable and self-relevant healthcare system in Asia region in July 2016. And later in July 2018, basic policy was revised to include the contribute to elimination of drug medical device lag in light of drug and medical devices indispensable role in healthcare. And the headquarters for healthcare policy established ground design for Asian pharmaceutical and medical device regulatory harmonization to improve access to pharmaceutical and medical device in Asia in June 2019. Next. This slide summarizes action in grand design. First action is to establish system and framework comprising various stakeholders. Based on the system and frameworks, we can enhance clinical trial system in Asia and promote international standards and capacity building with action specific for each product category, drug, medical device, and in vitro diagnostics, and regenerative medicine as action 4. EMDA Asia Training Center is also contributing to action 3, the promotion of harmonization through capacity building in Asia. So next, please. This is my last slide. Today, I share the Japanese experience on utilization of ICH-E5. Recently, utilization of bridging study is decreasing, but brought by ICH-E5 is still actively utilized, such as ethnic factors and extrapolation from a population to another population. Besides these concepts, we would like to create evidence in Asia, collaborating with other stakeholders. We believe Taiwan is the most important collaborator. We are looking forward to working with you more and more. Thank you so much for your attention.
Thank you very much, Dr. Sato, for your excellent presentation. And you share about the Japanese view, how to implementation of the ICH-E5. Okay, now I'm going to the next presentation in this session. It's on critical visibility, the city and its encounter with regulatory world. I would like to call on Dr. Wenhua Guo to present this subject. Dr. Guo is the profession of the Yangming Jiaoda University. She is a medical doctor and a graduate from the National Yangming Medical College. and received PhD in History and Social Study of Science, MIT. His research interest major in social study of medicine and pharmaceuticals, institutional approach to global health in East Asia, East Asian traditional medicine, the making of care professional in hospice. Let's welcome Dr. Wenhua Guo. And I believe we recall his presentation in advance.
Yes.
You will? Okay. Dr. Guo, please.
Good morning. My name is Wenhua Guo at National Yangming Jiaotong University in Taiwan. As a social researcher who worked on the ICH and East Asia in my early career, I'm grateful and honored to be invited to participate in this special occasion celebrating the 20th anniversary of Bridging Studies, one of Taiwan's Center for Drug Evaluation CDE signature policies. Just to show some photos I took in my fieldwork back to the early 2000s. This is photos from the ICH E5, the first D5 as per working group 995, among this some big names there, and ICH E5 as per working group dinner as in 2003. And a few experts I interviewed in Japan, from left to right, Dr. Chikaki Naido, Naido Sensei, and Mr. Osamu Doi, 2006, and age, which actually he helped me a lot to know the E5. And also I did some research the Taiwan city as well. This is the old CDE back to 2003. It was beautiful. It was beautiful. And on the right is a symposium of the APAC Network on Pharmaceutical Regulatory Science I took in 2007. So there are several good memories about it. But today, by way of summarizing the research I've done on this topic, you can see in the proceedings provided by the CDE that I have written several articles on this. In today's presentation, I would like to respond to the CDE's official website, which I listed here, which states that intensive international engagement with the ICH began in 2008. It's intriguing because it is the starting of the second decades of the CDE, and it's curious about what happened in the first 10 years. From the bureaucratic perspective, that statement is correct. The website lists the ICH Assembly since 2013 and marks the years of the CDE as an observer and formal member. Nonetheless, from the institutional perspective, it did not offer much of the sense of how Taiwan made it through to participating in global health via the ICH, that the most significant regulatory platform that facilitates the conversations between health authorities and industry at the frontier of global pharmaceuticals. So the goal of today's presentation is to replace some of the issues, events, and the people revolving around the first decades of the Taiwan CDE. As a professional, yet unconventional, government-sanctioned organization, the foundation of the early development of the CDE earned itself global visibility when encountering the regulatory world. It has not only built up an infrastructure capable of providing quality reviews comparable to global benchmark agencies, it also created robust connections with them as a means to catching up. This effort, I argue, prepared the institutional momentum needed for the ICH launch into the ICH among other stages of global health. What made the my above point more clearly. Some background on the trade negotiations between the US, Japan, and Taiwan in late 1980s and early 1990s has to be introduced, something I would like to call pharmaceutical diplomacy. Because of regulatory incompatibility between the US and Japan, then the world's two largest national markets for pharmaceuticals, Japan was usually accused of creating regulatory hurdles for the US-based pharmaceutical industry. In this context, pharmaceuticals were first put on the table for trade negotiation in the first US-Japan market-oriented sectors selective talks, or say MOS, in 1986, in which an agreement was made regarding the principles of accepting foreign clinical data. While working with Japan on the same topic, the European Union, later EU, also took the question of acceptability of foreign data to be an issue concerning whether integral regulatory platforms should be formed across member states. In the end, the ICH presented a perfect scientific solution for both regulators and the regulated. No universal standards can be achieved without others' involvement. At the same time, Taiwan was facing huge pressure to open up its market. From 1986, the same year, the pharma began to pressure the Taiwanese government through the Special 301 provision of the Omnibus Trade and Competitiveness Act, under which the USTR targeted trading market of the United States for their unsatisfactory protection of the US intellectual property. In the following years, an arbitrary bar of clinical trials was established. A requirement of 24 subject trials for bioequivalence was half of participants designated as a control group in 1989, followed by an even higher one of at least 40 subjects for every product that sought registration beginning in 1993. In hindsight, the ICH could have been a regulatory solution for such commercial diplomatic dilemmas, but in practice, this has not come about until the third ICH conference in 1995. So the regulatory and commercial concerns described above dominated the way accent differences were discussed at the ICH. This is the origin of the E5. Thinking that it was crucial to the acceptability of foreign clinical data, and arguing that a consensus on the issues should have reached on a scientific occasion, or some doing I introduced earlier in this presentation, then the representative from the Japan's Ministry of Health and Welfare to the ICH actively promoted this as a topic to be evaluated there. Upon its in the systems, it was agreed that race should be discussed among other differences in populations, such as sex and age, and an expert working group was thus assigned. Titled Accent Factors in the Acceptability of Foreign Clinical Data, this E5 topic took an exceptionally long 6 years of negotiation compared to an average of 1 or 2 years, during which 21 drafts were written, including a midterm report, so it's very, very long. There's no need for an exhaustive review of how differences among populations would deal with during the negotiations. Briefly, Western nations stress the fundamental unity of humankind and claim that further clinical trials should be added only if it could be proved that there's only real differences unique to Asians. On the other hand, taking for granted the genetic and cultural distinctiveness of its population, or under an umbrella term in Japanese, Minzoku, Japan insisted that no trial should be foregone unless the similarity between the Japanese and other ethnic groups could be proved. Two divergent proposals were submitted. While its Western counterparts asked for more waivers in the late-phase clinical trials, Japan expected the trial system was equal representation of whites, blacks, and patients feared Japanese. Neither of these proposals pleased all parties, resulting in a deadlock. So as you can see, then the rapporteur of E5 EWG, Chikayuki Naito sensei, recalled, I was ready to accept that there's no agreement was achieved. It was Roger L. Willems from the US FDA who saved this discussion by raising the idea of bridging study. Technically, bridging means undertaking extra studies to generate the necessary information for extrapolation of late-phase clinical data to the population of an untested region. In practice, nonetheless, it was a compromise to break the deadlock by leaving parts of the procedure ambiguous and open. From the viewpoint of the EU and US, the bridging study was a way of testing whether existing data could be extrapolated to the local region where the product was remarketed, and was only to be applied if the product was suspected of being accidentally sensitive. From Japan's viewpoint, however, the bridging study was considered a diplomatic result that allowed additional studies to be designed, especially for Japan, who made it as full study by using smaller Japanese samples. Although confusion remained, the guideline was agreed and upon in 1997 and implemented in a year later. And ambiguous as to how bridge studies should be performed suffers immediately after the implementation of the E5 guideline. Because of the ICH respected local agency's judgment as to whether a drug may have accidentally related effects, Japan was the first state to come under industry's scrutiny. Not so surprisingly, Japan's regulatory agency at the time, the Organization of Pharmaceutical Safety and Research, OPSR, or in Japanese TICO, exercised its authority to ask all procedures and producers to provide local clinical data for review and rejected those studies that aim to generate Japanese clinical data only to complement the application package. As you can see as a slide, it's some of the early slide in my fieldwork. You see you can see it's the foreign data had to repeat all the things with Japanese data. By the time the sixth ICH conference was held in 2003, only a handful of cases that claimed to apply the concept of bridging study had been accepted. Comparing it to Japan, what made Taiwan visible to the ICH was the way the ICH evaluated the differences among populations. While recognizing differences between Asian and other major races, it does not insist that clinical data produced from one population cannot be applied to another, even after careful evaluation. The CDE required bridging studies only when application was considered accidentally sensitive and welcomed Asian data from outside Taiwan. In addition to the consulting system, the CDE demonstrated its transparency by providing comprehensible flowcharts of applications and checklists for required materials. The results speak for the policy. By 2003, local trials had been deemed necessary for bridging studies in only 15 out of 62 applications, and the industry was praising Taiwan for setting out the workable model for bridging. Using the E5, the CDE turned its regulatory crisis into an opportunity. First of all, compared to major markets, Taiwan had been lagging in setting up its own regulations, and it was just a hard decision for the Chinese government to decide to rid itself of these. Moreover, it could even use ICH as a leverage to raise its own bar to meet global standards. Second, the CDE set a rather long period of regulatory transition. By 2004, a product sponsor was eligible to choose between conducting the 40-subject trial or submitting its package for bridging study evaluation. Third, The CDE hoped to prove its ability to conduct sensible bridging intentionally or not. Taiwan was characterized as an assembler of non-IC states, where engineered a win-win situation that enabled industry to make available and the latest medicine while protecting the health for its own people. The CDE's appearance on the map of regulatory science has orientations. As a non-ICH state, Taiwan would have been unlikely to affect the ICH discussions. However, the E5 guideline provided Taiwan with a concrete platform. In contrast to Japan's hesitant attitude toward the guideline, Taiwan immediately announced that it wanted to be the first non-ICH state in Asia to adopt it, including its controversial sections in 1998. In effect, the newly founded CDE took responsibility for the implementation of ICH guidelines and functioned as a liaison for all international affairs relating to the regulatory science in pharmaceuticals. It should make clear that the CDE's critical visibility was not simply gratuitous. As early as its meaningful attendance of the ICH back to 1995, 2010, and our host here, later the deputy director of the CDE felt that the E5 should have been a regulatory means to turn Taiwan into the ICH. And eventually he wrote a report for the Department of the Health in which he proposed a 3-year project to establish a committee to keep track of ICH actions. The opportunity to host the Drug Information Association DIA meeting in 1998 granted Taiwan the chance to make itself heard to the global regulatory world on its policy of bridging. This policy, as set up in 30 papers on clinical trials in the Asia-Pacific region, half of which were either written by Taiwanese authors or about Taiwan, was included in the special issue of Drug Information Journal, gets edited by Chen shortly after the symposium. The Network on Pharmaceutical Regulation and Bridging Studies under the scheme set up the Asia-Pacific Economic Cooperation Forum, APEC, founded in 2000, was an institutional platform that kept the CDE visible. So perhaps the most valuable achievement Taiwan made during this regulatory transition was the establishment of the CDE as a reliable institute. Aiming for the same high standard as the US FDA, its effort to introduce the ICH guidelines made the CDE a unique institution, unlike those in many non-ICH states, and unlike even those of the other governmental departments in Taiwan. To maintain critical visibility, the CDE kept balancing diplomacy and the science, biostatistics in particular, exciting its regulatory colleagues with a new idea from the mainstream. Here just list some of them. For example, the FDA's critical path and the EU's pharmaceutical evaluation reports scheme or per scheme. And when the ICH approached the People's Republic of China and India as emerging markets for pharmaceuticals in 2008, the CDE made its way to the ICH Registrars Forum, high and bright. Policy assessment always deals with a gap between what is ideal and what is real, and bridging studies are no exception. This does not fit well with our imagination of globalization. I think all the people here knows. And I quote here the John Lim of Singapore's Health Science Authority once suggested, if we're global, there will be no need for bridges. So I think our jobs here is try to make the two ends meet. Even so, as a working concept, bridging study has become the permanent part of regulatory repertoire, just like Taiwan's membership of the ICH. So what I quote here is by one of my admired senior scholar in my field called Murakami Yoichiro in his book Anzengen or on security. He said like any ultimate solution for all cultural variations and the differences is not invariably free from a particular viewpoint that channels all interest in a certain direction. So that is saying we do not want to see in the ICH. But luckily, it did not happen. Well, but as saying it's part of the routine, I will say as a policy researcher of health and society, I'm always advised that there's no easy ways for Taiwan, a de facto state that lacks legal recognition as such, to interact with others in international health affairs. The ICH, in this sense, is an unusual example. The early CDE's excellent performance and witnessed in the regulatory world deserve more attention. How it has managed to make its policies visible and referenceable has produced what I call a voice on the bridge. So that is maybe one of my most memorable papers on this concept to portray Taiwan's policy just as lights. I mean, this voice can be clearly heard in regulatory world. But still, this is perhaps the most important lesson we can learn from the early history of the CDE. The philosopher Isaiah Berlin used the metaphor of the hedgehog and the fox, in which hedgehogs view the world through the lens of a single defining idea, while foxes throw the wild variety of experiences. The early ICD fortunately owned the characteristics of the two animals. Holding a belief in science, erase its regulatory voice, and become the game changer in the heterogeneous world of pharmaceuticals, in which regulatory bridges are needed to connect the regime the one to the other. As the city's official website states, it had been intensely engaged in international participation since the second decades. That's true. How to agree with this evolving world, which Korea and the PRC also join as players, deserves another serious study. So I shall stop there. Thank you for listening.
Thank you. Please give a big hand to Professor Guo. Thank you very much for your insightful talk.
So can I say a few words?
Thank you. Now, ladies and
Can I say a
gentlemen.
few words?
Sure.
Yes.
Absolutely.
Okay, thank you very much, because I only took about 22 minutes, so allow me like 5 minutes to say big thank you to give me the opportunity to talk to all of my admired colleagues, and also give me the chance to speak myself. I want to express my gratitude to Dr. Liu for the kind introduction, and I want to express my deep apology in that because of the COVID pandemic that prevent me from being here to join these admired colleagues. And I would like to say a few words because I personally like Professor Oliver, whose comments on like who is a first runner, who is a guy, who is a country that make history and allowing others read history. But here, I strongly feel that it's a specific moment that we have to recall history, and this history just comes in a very, very hard time as for now, COVID. And I just like to share with you some personal experience as a researcher on this topic. That was back to 2003, and that was SARS. So I'm so grateful for the people who introduced me to the regulatory world and allow me to say what I want to say, and then discover a lot of stories. I mean, in addition to the Japanese senior researchers that I have introduced in my presentation, I also want to add a few names from Taiwan, including, of course, Mr. Bridges Studies, Dr. Chen, and also I have to say my deep thanks to the former the CEO, Professor Jumon Lin. Actually, Jumon Lin introduced me to Dr. Chen and very kind to me when we met in the very difficult time of SARS. And additionally, there are several people I think I should give some mention that include my professor Huang Wenhong, and of course Professor Oliver Hu, and also Dr. Xiao T. Chen and Dr. Chen Kuan-chi. I think these two Chens actually helped me know a lot about what's going on there. And of course, I have some contact with Yijie Lenmong at the time, and I want to express my thanks to Wu Shuming. I will say like this generation of physicians like to have a broad vision about what's going on and what we can do outside of clinical field. So inspired by Dr. Chen, Dr. Wu, Dr. Many, a lot of seniors, I have to say like now we can see the great, CD or say like the CDC and some of the physicians, officers, working on this. I will say like, it's so grateful to be part of this great tradition of physicians involving in global health and public health. So just a few words, I feel so moved. So I would like to share. Thank you.
Thank you very much, Professor Guo. Thank you. Will we hear more from Professor Guo? Because now we're going to proceed to panel discussion, and then it is our great pleasure to have Mr. Kevin Mingxun Liu, Chief Executive Officer of Center for Drug Evaluation, and Dr. Hengde Chen, Distinguished Research Fellow of Center for Drug Evaluation, to moderate the session. Now, please welcome them onto the stage. And we also welcome our panelists of the session, Ms. Ting-Ya Chang, Section Chief Chang, to come up to the stage. And for Dr. Junko Sato and Dr. Wenhua Guo will also join our panel discussion live via video conferencing. And now, if the floor is open to the floor, to our participants here on site, please raise your hand and our staff will hand you the microphone. Please state your name and your organization before raising your question. Now the floor is yours, please.
Okay, thank you. Once again, I want to thank you, Dr. Guo, for your excellent speech, and you a lot of kindly comment for CDE, and I received another message, and I and my colleague will keep ways toward the global, and you mentioned aim to be rule maker rather than a follower. I received that message, and my colleague will do our best. Now, because Dr. Hengde Chen joined us, so we welcome your question and comment.
Thank you. I think we have a very exciting talk from both several speakers and also from Dr. Junko Sato and Dr. Wenhua Guo, that we know that throughout the BSc process, actually there are a lot of timing from ISO E5, from Japan, from global company, from Taiwan initiative. So all this put together, then we have this 20-year BSc. But looking forward, I think in my last slide and also in Dr. Junko Sato's last slide, talking about the future Asia pharmaceutical and medical device regulatory harmonization, I think that's quite important because I talk about the timing is right. FDA starting to look at how they can have a clinical trial with ethnic group, racial group represented in the United States. And so I would like to ask Dr. Sato first, before open to the floor, that is there any plan that we can work together, especially when I mentioned that BSc checking self-assessment aid could be a tool that we work together and we share our comment on the concept of ethnic group in that particular drug and indication as a starting point that we can work together? Because I know that Japan is also trying to join the FDA project office. I know that you are like shadowing under that project, and that project also heavily use assessment aid. So I wonder if Dr. Sato want to comment on this. So I would like to hear a comment from Junko Sato.
Dr. Sato, can you hear us?
Yes.
Did you-
Okay.
Okay.
Thank you so much, Dr. Handa Chang, asking it to me. So I think multi collaboration is very important. So as a regulator, we are promoting the regulatory harmonization and communication to know each other. For example, between Taiwan and Taiwan FDA, CDE, and Japanese MHLW and PMDA has a regular communication to share regulatory update and to share issues to resolve. And in addition to such regulatory communication, academia collaboration is also very important. So currently, Japan is promoting clinical network in Asia. So if academia has a communication to conduct clinical trial, not only for regulatory approval, but also to generate evidence in Asia. So communication in academia is also important. And another point is, I think it is very important, but a little bit difficult to resolve. I think that communication in industry. So usually we ask global pharmaceutical company, please start clinical trial in Asia at first, not the US or EU, but usually companies say, so at first they would like to start EU or US of their clinical development. So such, how to say that, development strategy in company has been changed. I don't know, good idea, but if company's concept is not changed, so our problem is not solved. So, but I think such collaboration in academia, regulator, industry, very important. So I don't know, Japan can how to contribute the problem, but we are trying to resolve this problem and promoting the collaboration in each academia, industry, regulator, etc. Thank you.
Oh, thank you. So I think we all face the same problem, how to change the current situation. And Taiwan has done a little bit through our bridging study, self-assessment aid. So hopefully, perhaps Japan or future United States, US FDA can, based on this concept, to develop their own assessment aid, then we can better communicate on this ethnic factor sensitivity. So I would like to open the question to the floor or the online. If anyone have any question, yes, please. Dr. Chen.
Okay, it's Ming Xiao Chen from CD Taiwan. I'm the Division Director of New Drugs. And I have a more practical question to Junko Sato-san. Yeah, thank you for your sharing. And as a reviewer, I would like to know, for orphan drugs used to treat rare and serious disease, And regarding bridging study evaluation from regulatory perspective, I would like to know, does PMDA take a different approach or probably more flexible approach for drug approval, even with very limited Asian or Japanese data? Thank you.
Thank you for your question. So regarding orphan drugs, so yes, we have a very flexibility. So for example, I think Gaucher disease, so we approved a drug for Gaucher type 2-3 disease. The number of patients is very limited, less than 10 in Japan. So at that time, we review the effect of drug by change of clinical symptom, etc., in a patient. So it is not clinical evidence, but generation of evidence, generating evidence is very difficult for orphan drug. So we usually review the efficacy, compare between pre-administration and post-administration, how to change the symptom, etc. Then, regarding such drug, communication with patient and the caregiver, physician is very important. So why PMDA MHLW approved this drug? What evidence, what data is available? So what are unknown? So we usually share such information with patient, healthcare professional, and caregiver, usually family, etc. So then, so we usually ask informed then to patient before administration of drug. After approval, we ask MAH and also Association of Physicians Group to collect post-approval data to collect real-world data. So these are honest situation regarding orphan drug review and communication with patient, etc. Thank you.
Online, we have 3 questions for our section chief, Yating Zhang. And one is asking slide 8 about the bridging study criteria. And the other is asking about the e-consent of e-informed consent for doing clinical trial. Is that applied in TFDA? I think that a special announcement during the COVID-19 in the slide 27. The third one is that for company want to do first in human clinical trial, if they already have the trial approved by one of the 10 reference country, do we have a accelerated approval process? So, perhaps section.
Thank you. About the question 1, my answer is the data meets bridging study criteria means that the data provide pharmacodynamic or clinical data on efficacy, safety, dosage, and dose regimen in the new region that will allow extrapolation of the foreign clinical data to the population in the new region. And about question 2, is electronic consent forms acceptable to TFDA? My answer is, in principle, using electronic consent form is also allowed in Taiwan. If there is company who want to use electronic consent forms, it is needed to follow the announcement for e-consent in 2017 by Department of Medical Affairs. The consent process and the confidentiality of information should still follow GCP regulations. And about question 3, what kind of review process will it be within a first-in-human clinical trial, which has approved in one of 18 countries? Is there fast-track process? My answer is FIH, first-in-human clinical trials, could not go throughout for MRCT fast-track process. But if there is FDA approval, it can go through fast-track IND process for a new drug with IRB approval. Thank you.
Thank you. Any other question from the floor or from online? Yes, please, Dr. Xu.
Yeah, I'm Li Juanxu and work at CDE, and I have a question for Dr. Sato. And thanks, Dr. Sato, for your excellent presentation, and I'm glad to hear you discuss about the similarity in Asia and Asian cooperation. Based on these thoughts, is it possible, instead of Japanese data, PMDA accept other Asian data, for example, Taiwanese data as bridging data? Thank you.
Yes, please.
Yep. Thank you so much for your question. And yes, we can accept foreign data. So, but we usually require also Japanese data, because sometimes especially not efficacy, but safety profile are different between Japanese and other region. So therefore we required usually Japanese data, except for special area, for example, so HIV drug, etc. Yes, and so, but we can accept we welcome foreign data, especially in Asia, including Taiwan and Thai, ASEAN data, etc. Because if we accept such Asian data, the development period will be short, and will shorten, and also maybe decrease development cost, etc. We can expect such things. So we encourage industries to conduct MRCT Asian clinical trial, so to obtain such benefit. Thank you.
Okay. I have an interesting story, because about 10-some years ago, Taiwanese company asked me, will Japan accept pharmacokinetic data from Taiwan? Many say, no, you have to do it in Japanese. But just never clearly said in public. So I wrote a letter to PMDA, Dr. Daiichi Sato, who is responsible for this, and also a good friend of mine. I said, do you accept Taiwanese pharmacokinetic data? And he wrote me an email. He said, yes, we accept, and we have examples that we already accept. So I say, anyone who question this, then I can show the person this email. But I think safety probably have some extrinsic factor, but pharmacokinetic, since 10 years ago, PMDA said they accept Taiwan PK. But I think that's not also still not very clear to many industry friends. Yeah. Any question? Dr. Sun?
Thank you very much for Dr. Sato's excellent presentation. Just following the previous questions, I don't know if in Japan, do you have any experience about when you review the PK data, is there any difference between Taiwanese, if you have Taiwanese data, and any difference between Taiwanese PK and Japanese PK data? Can you share about this?
Yeah, thank you for your question. I don't have such. I'm not sure in other region, but other therapeutic area, but in anti-infective, I don't have such experience. It means, usually in Taiwanese and Japanese has a very similar PK and drug response for efficacy, etc. Yeah. Thank you.
Yeah, so not many experience, but the few experience seem similar. And certainly it's a good reference, but whether that can be enough, that can be discussed. I think perhaps also a case by case. Yeah. Any further question?
Any other questions from the floor?
And also, I would like to ask Dr. Wenhua Guo, because we have a very interesting encounter in 2003, that suddenly you contact me through Dr. Zhu Monglin, said you want to do a PhD dissertation on bridging study. How you come up with this idea? How you find this topic? Because we never met before that.
Well, thank you for the question. I will say, it's a terrible thing to have a good memory, and you always allow me to think about these good old things. Actually, 2002, I was in Todai. So I attended a conference about the early development of the negotiation between Japan and the United States at the time. And then I have some friends with my professor at Sendanken. I don't know how to say that, but something like a research institute attached to Todai. So actually, I know Taiwan from Japan. And then, Japan is always a good reference for me to know the different strategies to cope with asset differences. So this is a brief answer to that. I will say it was a surprise because because at that time, my focus is between the US and Japan. So actually, I wrote an article with the two Japanese scholars on the harmonization of regulations. It was published in the DIA. So in that article, I focus on the role and the function of DIA in the early promotion of Japan's relationship with other countries in terms of regional harmonization. And also, I got a chance to know some of the early plans of the global drug development. So I will say why I call my presentation the critical visibility. I have to say Japan and Taiwan should work as a couples because without Japan, we cannot generate any critical visibility. Thank you.
Okay, thank you. And any more question or comment before we close this section? If not, can I invite Dr. Sato and Dr. Guo say a few words for us before close this section. Any comment, any more comment or suggestion for us?
Okay, so can I just say a few words, because I would say like I should leave more time for Sato-san. I would like to borrow this place to thank all of my Japanese colleagues like Mori-san or Sato-san, and another Sato-san, and a lot of good people, especially Dr. Naito. Dr. Naito is a pioneer of this field. And also, of course, Uchida-san. Actually, I have an article in Japanese on the very tough negotiation between Japan and the United States. That was published in a book by Todai Shobankai. So I think that will be a good reference. If you have any symposium like this, I would like to share my experience with my Japanese colleagues. Thank you.
So thank you so much, Dr. Guo-san. So yeah, I saw many old friends in your presentation today. So Doi-san, Dr. Naito, so etc. So I when I watched Dr. Guo's presentation, we have a long collaboration, communication history between Taiwan and Japan. So we would like to, we already have a good communication collaboration, but we would like to promote more and more to provide innovative product. So unfortunately, last a few years, we can't meet in person due to the COVID pandemic, but we recently we restarted to go to abroad. So we restarted more communication in person and to tackle new issue, for example, regenerative medicine and etc., and to provide, to utilize them as a technique and also as a new pharmaceutical product. As I said in my presentation, Taiwan is the most important collaborator with Japan. So we would like to keep in touch and collaborate and work together moreover. Thank you so much.
Okay, I think it's about time to close this section. And once again, I want to thank you, Dr. Sato, Dr. Guo, and the section chief Zhang, and still online our participation. And now we close this session. Thank you.
Thank you. Please once again give a big hand to our moderators and our panelists. Thank you very much. And we thank all the participants. Thank you very much. Welcome back to the conference. We will now commence the third session. And now please join me in welcoming the moderator of the session, Dr. Jiaxin Ye, Director of Division of Pharmaceutical Science, Center for Drug Evaluation, onto the stage. Welcome.
Okay, ladies and gentlemen, welcome back to the conference. Good afternoon. I am Jiaxin Ye, the Division Director of Pharmaceutical Science in GDE. In the afternoon, Director Zhang and I will moderate the afternoon 2 section and with 5 speakers. We will talking about the bridging study variation regarding from regulatory perspective as well as from industry perspective. And in my section, I would like first introduce our first one speaker is the Mr. Jizhen Yang. Now, Mr. Yang is get his BS degree in life science in National Central University, and his master degree in biochemical science and technology, National Taiwan University. And then he goes into academic silicon as a research assistant. After his job, he joined the CDE at the current position. He is now as a project manager in Division of Regulatory Affairs and Compliance in CDE. Today, he will give us about the talk of an overview of logistics and analysis of bridging study evaluation application. Welcome to Manager Yang.
Thank you, Dr. Ye, for the kind introduction. So I'm Qizhen Yang. I'm project manager at CDE. So I actually kind of relatively newbie at CDE, you may call. So I'm really grateful for this opportunity to be here. And it's my great honor to present you an overview of logistics and analysis of bridging study evaluation applications. Oops. Next slide, please. Okay, so in my presentation today, I will walk through some background and regulatory progress of bridging study evaluation in Taiwan, and also what's the current regulation and review process. And I will also share some analysis of the BSc applications that we reviewed. Finally, as the conclusion and some future prospect. And I want to, before going into the details, I want to first apologize that if later in my presentation you may find some contents similar to morning sessions. And for those contents, I will either skip them or I will walk through them more quickly. Next, please. Okay, so please let me take you back to like 30 years ago. And if you remember, Professor Guo mentioned that there are trade negotiations among the US, the Japan, and the Europe. So back to 30 years ago, there's a lot of improvement of drug patent protections in Taiwan. So we had a lot of meetings and negotiations with the America, and like we have a economic council in 1992, and later we have an agreement of the protection of copyright with the Americans. So eventually, these backgrounds and these events lead to the July 7 announcement in 1993. So the purpose of this announcement for the Central Health Authority in Taiwan is to protect the pharmaceuticals innovations. In addition, the authority also wants to establish a solid and sound pharmacovigilance system and to improve the development of local clinical trials in Taiwan. Next, please. So the contents of the July 7 announcement is twofold. First is about the new drug application. So for the first time at that period, the applicants should provide the local clinical trial reports. And second is after the NDA issuance, there's a 7-year period of drug safety monitoring, which consists of 2 stages. First one, after the issuance date, there's a 5-year when there are generic drug sponsors, they want to apply for ANDA, they should provide local clinical trial data. And those data must meet with the same standards to NDA holders. In the second stage, the last 2 years, generic drug sponsors, instead of local clinical trial data, they need to provide bioequivalence study reports. And in addition to those administrative protective measures, during the safety monitoring period, any medical institutions like hospitals, they cannot ask for the clinical trials or trial use of those individual drugs. last but not least, for the NDA holders, they should provide their drug safety report to the health authority periodically, that is every half year. Next, please. Okay, so put those improvements aside, even though we have created a more innovation-friendly environment, there were still criticisms arose because the regulation required the applicants to provide local trial data with at least 40 subjects. But with only 40 subjects, you may know that it is hard or difficult to prove any clinical or statistical significance. So those criticisms pushed us to be more in line with international regulatory trends. Next, please. Later years, the ICH E5 R1 was released and finalized. And you may remember, or you may recall that Drs. Guo and Dr. Hu and Dr. Sato also mentioned the ICH E5. So I will skip this to the next slide. Thank you. And essentially, this guidance defines what is a bridging study, and it provides a general guideline for the regulatory agencies around the world and also the industry how to use a bridging study to accept foreign data. Next, please. So based on the ICH E5 and the above events, the Central Health Authority in Taiwan announced Double 12 announcement in the year of 2000, which is based on, again, ICH E5, and also to avoid unnecessary waste of R&D resources and to further elevate the standards of local clinical trials. So from now on, the NDA applicants, instead of providing local clinical trial data, they are now required to provide bridging study protocol or bridging study report. And there's another option for them, that is before an NDA applications, applicants can submit bridging study evaluation first with complete clinical data package, which should include Asian data. And all the BSc reviews will be based on the ICH E5. Next, thank you. So we have made a lot of progress over the past 20 years. And here shows you a summary of that. And please note that the green lines indicate international regulatory progress, more specifically for ICH. And the magenta lines represent the domestic advances. So I will jump to the middle part, the year of 2009, where the subsection 1 of Article 22 of Regulations for Registration of Medicinal Products was amended, added by the Central Health Authority, in which the drug items and categories that are subject to BSE were announced. And during the same year, the July 7 and Double 12 announcements were revoked, since the BAC regulations are now into the article. You have also heard about the E17 guidance released in 2017. And later in 2019, TFDA announced that drugs treating pediatric or rare severe diseases are exempted from BSE. And later in the last year, the article, the subsection 1 of Article 22 was amended to include those drugs, and in addition to put that cellular and gene therapy products are also exempted. But please note that the ethnic differences will still be evaluated during the NDA review. And in 2021, there's also an amendment that instead of Asian data, East Asian data should now be provided within the CCDP of the BAC packages. Next, please. Okay, so what's the current regulation and review process? So according to the article subsection 1 of Article 22, new chemical entities and new biologics need to submit bridging study evaluations. And when can you submit those applications? You can submit before your NDA, or you can together submit with your NDA. And applicants should prepare your package based on the BSc checklist, and you should provide CCDP with data for East Asian population. And I will skip the exception parts. So next, please. Okay, so many of you may be familiar with the evaluation process, but I want to emphasize that if the applicants have any doubts or concerns with your data, or you just want to have someone from the regulatory perspective to discuss with your developmental strategy, you're more than welcome to consult with us, with the CDE. We can definitely help to solve your problems. Okay, so back to the application. The application fee is now raised to $100,000. And based on the review results, an applicant would either get a bridging study waiver or a rejection. And if a rejection notice is issued, then a bridging study might be needed. So, applicants should prepare the package based on the BAC checklist. So what's about it? An applicant should provide your drug's worldwide clinical trial status. Your CCDP should include either NDA expert report or an investigator's brochure. In addition to clinical data from East Asian populations, applicants should also provide its comparisons with other ethnic groups. And there's a part of checklist called self-evaluation. And in that, applicants need to describe your drug's characteristics, like is your drug sensitive to ethnic factors? Are there different indications and or epidemiology? And applicants also should to summarize whether your drug is of any clinical or risk-benefit impact, such as whether indications are for serious disorders, etc., etc. Next, I want to share some analysis of the applications that we have reviewed. But before that, I want to share the applications number. So over the past 21 years, the CDE have reviewed more than 900 applications. So that's around 44 cases per year, shown in the left panel. So the majority of them are imported drugs, about 89%. And there are 11% for domestic drugs. And if we separate them into drug categories, the majority, about 67% of them are new chemical entities. And the second most is non-NCE. And the remaining 50% belong to the biologics. So how about waiver rate? So again, we separated the applications into imported drugs and domestic drugs. Over the past 1 years, there are more than 800 applications for imported drugs. And there are 577 got a bridging study waiver. So the waiver rate is around 70%. For domestic drugs, there are more than 100 applications, and the waiver rate is around 58%. Overall, the waiver rate is around 69%. So I also want to share with you the waiver trend. So here shows you the annual waiver rate from the year of 2011 to last year. And please note that the brown line indicates the actual annual waiver rate, while the green line indicates the linear trend. Before the year of 2011, the data I didn't show, but the average waiver rate is around 60%. From 2011 to 2016, the average waiver rate is around 70%. In the last 5 years, the waiver rate goes higher up to the average at nearly 90%. But as long as it's not 100%. That means there are still some deficiencies that eventually may lead to a rejection. So I will share with you some of the common deficiencies that we found among the non-wave applications, first for the clinical part. So the most common one being that the applicants provide not enough East Asian populations data, or the data is not sufficient to evaluate whether there are ethnic differences exist. The second one being that the provided CCDP cannot support the proposed indications in the BSE package. The applicants may have designed improper endpoints, or they provided trials but with different designs. Third, there are actual different indications or epidemiology between the East and the West that may also lead to any rejection. The fourth is the high-grade adverse events rates were discovered and that were different from the East and the West. Finally, if the long-term efficacy and safety data was insufficient, then that may also lead to a rejection. So now for the PK/PD part, the first common one still being that there is not enough PK data from East Asian population. The second one is the data cannot clarify whether there are PK differences between the East and the West. Third, there are actual ethnic differences of PK between the East and the West. Then some of you may remember that there's a part of self-evaluation where applicants need to describe your drug's characteristics. So if your drug exhibits like nonlinear PK, or it has a narrow therapeutic dose range, or its bioavailability has high inter-individual variation, that may also cause a rejection. Finally, if the data provided cannot elucidate if there's any drug-drug interactions, the mechanisms are still not clear, or the metabolism and excretion of the drugs remain unclear, that may also eventually lead to a rejection. So finally, we're going to the conclusion and some future prospects. Okay, so you may notice that, and I also mentioned in recent years, we have higher waiver rates compared to some old ages. So we have noticed that applicants actually now, they all can provide Asian data. So we wonder how many of them, in addition to Asian populations, they did provide Taiwanese data. So we separated into two groups, and we analyzed applications from the year of 2018 to last year. So in the upper row, you can see that CCDP provide Asian populations, but within those Asian populations, there are no Taiwanese data. And there are 67 cases, and the waiver rate is around 87%. So how about when the CCDP are with Taiwanese? There are 69 cases, and only 2 got a rejection. So that equals to nearly 100%, 97% to be exact, the waiver rate. So the result actually echoes, you may remember or you may recall that Chief Secretary Zhang mentioned in the morning that when the BSE package was provided with Taiwanese data, it can definitely help your BSE review to help the reviewers to determine if there are any ethnic differences. Next, we wonder how about the following NDA status of those BAC applications provided with Taiwanese. So here shows you the results of the 69 cases. 56 of them have their review finished and they got their NDA approval. And there are still 9 cases are under review, and there are 2 applicants have yet to submit their NDA package. And we have 2 cases the applicants eventually withdraw their cases. And the reasons are as followed. For one, that the reviewers found that there are safety concerns of cardiovascular risks, and the applicants eventually decide to withdraw the case. And for the second case, that the reviewers found there are efficacy and safety differences between the East and the West, so that eventually the applicants also withdraw their case. So since many applicants can provide Taiwanese data, that actually implies during their drug development strategy, they have started to include Taiwan for conducting their clinical trials. So we wonder, how about the multi-regional clinical trials number? Has them increased in recent years? And here shows you a plot similar to Chief Secretary Ms. Chang presented, that during the last 5 years, our numbers have been increased in both the IND and the CTN. So in this slide, I want to emphasize or focus on the CTN showing blue. So for clinical trial notification, or CTN in short, that are multi-regional clinical trials regulation for applicants. So if the NRCT trials meet the 2 criteria, then they can apply for CTN in Taiwan. So the criteria are listed below, that if your trial have been approved and conducted in one of the 10 reference countries, and if your clinical trials is planned to include one of the medical center hospital in Taiwan, then you can apply for a CTN. And why I want to emphasize this is because you got a very shortened review time. So you can get your results or your IND approval in just about 2 weeks, 15 calendar days to be exact. So even under the stringent circumstances of COVID-19 pandemic, in the last 2 years, we still have a strong growth in the numbers, both in the IND, but also in the CTN applications. So the number of the IND increased, we observed also across all phases. Phase 1, phase 2, phase 3, we have observed there are strong increase. Next, please. So finally, we know that when Taiwanese data were provided within CCDP, they not only help facilitate the BSc review, but also enhance its waiver rate and facilitate its NDA approval. And after the ICH E17 finalized and released, we have noticed that sponsors have been starting to design NRCTs, but also considering ethnic factors among different regions. And I just took that the number of NRCT trials have been steadily increased in Taiwan even during the COVID-19 pandemic. And finally, I sincerely encourage all the sponsors here to consider Taiwan for conducting your clinical trials, as we have world-class hospitals, excellent professional PIs, and COVID-19 management. I believe all of these have made Taiwan an excellent place for conducting clinical trials. So when you're planning conducting trials in Asia Pacific, we strongly welcome you and encourage all sponsors to include Taiwan. Thank you.
Thank you, Manager Yang, for the excellent presentation. And if you have any question, if you are online, you can type in the online chat room, and we will panel discussion. Okay, let's move on to the next speaker, take into the technology PK consideration. Our speaker is the Miss Yiling Wang She is our senior PK reviewer in my division in CDE. And I introduced his, he gave his BS degree in pharmacy in Kaohsiung Medical University and a master degree in pharmaceutical in National Defense Medical Center. And then he joined the CDE from primary reviewer, PK team leader, and the senior reviewer. So today he will talking about pharmacokinetic consideration of bridging study evaluation and the case study from regulatory perspective. Let's welcome reviewer Wang.
Thanks to the introduction of Dr. Yan. Good afternoon, ladies and gentlemen. I'm Wang Yiling. I'm glad to have this opportunity to share my thoughts about pharmacokinetic consideration of bridging study evaluation and case studies from Taiwan regulatory perspective. This is my outline. I have divided my speech into 4 parts. The first, introduction of BSE, then PK consideration, case studies, and the last, database analysis. Previous speech has mentioned a lot about ICH E5, E17, and even the BSE checking list. So I would like to introduce directly to how to prepare a BSE submission package. The first part, worldwide regulatory status, including the strategy of the product development worldwide and in Taiwan. And the second, the registration status in top 10 advanced medical countries, especially in Asian countries. For example, if the product was approved in Japan, sufficient clinical experience in Japanese population was expected and predictable. And then the most important is the list of all planning, ongoing, and completed trials. It could help all the review team to check the bridging data and decide the BSE strategy. The second part, CCDP, include a proposed indication, dosage regimen, module 2 or module 5, sure, the PK data in a new region. And all this selected information should have their full report as a supporting. The third part, a bridging data package is the most important part in BSE. According to Taiwan Regulation of Registration of Medicinal Drug, Article 22.1, it mentioned that when applying for BSE, manufacturers should fill out a checklist and provide CCDP, preferably with data on ethnic group in East Asia. So this means bridging data package preferably contain East Asian PK and clinical information. From PK perspective, we expect conducting PK and OPD comparison between East Asian versus non-East Asian. East Asian could be presented as pooled East Asian or grouped by country. For example, Taiwanese, Korean, or Japanese. And non-East Asian could be presented as pooled non-East Asian, Caucasian, or whole population. If the comparison was conducted in patients, and this should be representative of the major population in pivotal trials. Why we only accept the East Asian and not the data from all regions of Asia? It's a similarity analysis from two principal components across 30,000 SNPs. And let's first look at the left figure. It's from Asia. And the red point is Japanese, and the purple was Chinese population. And the left quarters were people from India, Sri Lanka, or Middle East. As you can see, the population in India was quite far from Chinese population. And that's why we don't think Indian was Asian people. And we usually require excluding Indian population from bridging data. And this population were from Malaysia. Sometimes the data from Malaysia was suspected because their ethnic composition was more completed. Okay, and then let's look at the right figure. The y-axis was scaling down, which means the population was more closer in genetic origin. The left quarters were minority and indigenous population from Chinese. So as you can see, the people from Chinese, Korean, Japan, and Okinawa, they were very close in genetic origin. So we not only accept Taiwanese data, we also accept all these data from East Asia as a bridging data. And the next slide was the main part that I want to make today. It's about how to select PK/PD information and how to present this information in your package. If the drug development enrolled East Asia in early phase with the timeline, in early phase, we can conduct a PK difference between East Asian and non-East Asian. As it comes to phase 2, we can conduct a PD difference between the two races. If there's an ethnic difference existed, you may can observe a dose response curve shift in East Asia. In phase 2, phase 3 trials with PK, spalling PK sampling, population PK analysis will be conducted. And subgroup analysis of race on clearance and volume of distribution could tell us the impact of race on PK. But it also depends on the sample size of Asian population. If the sample size was too small, it's hard to draw a comment on ethnic issue. But if the sample size of Asian population was sufficient, and you can further conduct a simulation PK between East Asian and non-East Asian, and they could be one of the evidence in bridging. For bioproducts, you should further consider immunogenicity comparison, including the incidence of anti-drug antibody and its impact on PK, efficacy, and safety. For pegylated product, anti-PEG antibody should also be included. Okay, and then how to present this selected data. The first, cross-study comparison. You can compare this data between or among 2 or 3 different studies. They should have a matching study design, the same study population, using the same formulation, in order to reducing the data variation. Or it could be a head-to-head PK/PD study between East Asian and non-Asian. It used to be a local trial or multi-regional clinical trials. And then PAPI-K, we just mentioned. And the critical elements is a representative of East Asia. As I mentioned, India was not acceptable, and the people from East Asia could be a bridging data, and their sample size. And the PK data from target population is more important than the data from healthy population. The PK from multiple dosing PK is more important than the PK from single dose, especially for repeated clinical administration drug. And the East Asian data should cover the maximum proposed therapeutic dose range. And all I mentioned, this selected information should have their full report as supporting. Okay. And these properties of a compound likely to be sensitive to arsenic factors is also a checklist on our checklist that you should clarify all these properties. First, linear PK, a steep peak deep curve for both efficacy and safety, which means a small change in PK results in a large change in efficacy. A narrow therapeutic dose range, highly metabolized, especially through a single pathway, and increasing the potential for DDI. Metabolism by enzymes known to show genetic polymorphism, a product with potential for ethnically variable conversion, high intercellular variation in bioavailability, and the last, low bioavailability with great food effect. People might question, what if the food effect from different cuisine, especially for drugs, should be taken with food? Since the food effect, a guidance of food effect was published by US FDA, the content of calorie, fat, protein, and carbohydrate was standardized. So with a clear recommendation in labeling, I think it was not a concern. I want to pay particular attention on this property. Metabolism by enzymes known to show genetic polymorphism. This is a table summary from US FDA. It's a pharmacogene genetic difference and the labeling recommendation. The first column were pharmacogene which were known to have genetic polymorphism, like CYP2D6, 2C19, UGT1A1. And this is the frequency of a poor metabolizer among these races. And the right part was the drug which was known to detect poor metabolizer and their recommendation in labeling. For example, for CYP2C19, the frequency of poor metabolizer was higher in Asian as compared to white and blacks. On the other hand, clinical response difference and safety difference was observed in these two drugs. So there are dose reduction and safety monitoring was required in the labeling, especially for poor metabolizer. We know that not all the genetic polymorphism reflect their difference in clinical, but in some drugs, they did observe some difference and due to the different rate of metabolism in the poor metabolizer. So with the technology of diagnostic of a poor metabolizer and the clinical and clear recommendation in labeling, I think it could be the solution about the genetic polymorphism. Okay. Have you ever wondered what drug class likely to existing arsenic sensitivity on PK? There's a table of a PK of a drug which was known to show PK difference between Asian and non-Asian. This is a drug column, and this is their pharmacological subgroup based on ATC classification, and the enzyme responsible for metabolism, and then the substrate. The top three drug classes were direct-acting antivirus, protein kinase inhibitor, and then lipid-modifying agent. And rosuvastatin was the most classic example in ethnic variability. It was majorly metabolized by CYP2C9, and it's also the substrate of OATP1B1 and BCRP. Let's look at the profile of rosuvastatin. Following a single 14 minutes rosuvastatin, the C-mesma and AOC increased twofold in Chinese population as compared to white population. And this difference could be explained by the higher frequency in Asian, the higher frequency, allele frequency of OATP1B1 and BCRP, and the higher allele frequency leading to a decrease in the function of these two transporters. So let's look at the right figure of hepatocyte. BCRP, OATP1B1 on hepatocyte is an influx transporter, and BCRP is an efflux transporter in hepatocyte. With decreasing the function of these two transporters, it could accumulate more rosuvastatin in blood. So that's why higher concentration in Chinese population. And I'd like to expand this idea to this drug. The same phenomenon was also observed in direct-acting antivirus. This drug was majorly metabolized by CYP415 system, and most of this drug were transporter of OATP1B1 and BCRP. This might be due because of a lower CYP3A4 contents in Chinese, a smaller liver volume in Asia, and lower OATP1B1 hepatic uptake activity in Chinese population. And actually, most of this drug was indicated for chronic hepatitis C. And most of this drug accumulate more in liver than in plasma. So consider liver is the most distributed organ in body, and also consider its pathology. I think it worth more discussion and investigation in this issue. Okay, and then a case study, Capresa. The active ingredient, bendetanib, it is a receptor tyrosine kinase inhibitor in patients with locally advanced MTC. The dosage regimen was consistent in Taiwan, Japan, US, and EU region. 300 milligrams once daily, taken with or without food. The bridging data include a cross-study comparison PK in cancer patient and PAR PK in target patient. The table is a single-dose PK. And all these 4 studies were conducted in cancer patients. I've divided this data into 2 parts. The blue circle were 2 studies using the same formulation, phase 1 formulation. And the orange part was a study using the commercial formulation. So under the same formulation, we can see the AUC and C-mesma in Chinese population was higher than that in Caucasian. And let's look at the multiple dosing PK. It's also the same study. Following the therapeutic dose, 300 milligrams once daily, the C-mesma of Caucasian was almost 1,000 nanograms per milliliter. However, for Chinese population with repeated administration of 300 milligrams, the C-mesma as high as 2,000 nanograms per milliliter. And the situation also can be seen in AUC, twofold difference. Okay, and let's look at the PD result. In Capresa, there's an important safety issue. A substantial, sustained, and concentration-dependent QTc prolongation was observed in Capriosa. And so let's look at the figure. It's a PK/PD curve from Caucasian. For Caucasian, following a 300 milligram once daily, the CMSMA was located in 1,000 nanogram per milliliter. And so there's a 35 milliseconds of a QTc change from baseline was expected. However, for East Asian, if there is Asian receiving the 300 milligram, the CMSMA was located in 2,000 nanogram per milliliter. So more than 40 milliseconds change from baseline of QTc was observed and what was predicted. And indeed, the safety issue was observed in clinical. So higher incidence of a QTc prolongation was observed in East Asian in clinical trial as compared to Caucasian, 8% versus 4.5%. So because of the safety concern, as compared to global version of labeling, we required additional warning in the labeling of Capriosa in Taiwan. As highlighted in yellow, we can see there's a post-marketing requirement. And we also mentioned that in Capriosa clinical trials, higher systemic exposure and higher incidence of QTc prolongation was observed in East Asians. And the optimal dose for East Asian population still remain unconcerned. So to sum up, the BSE and the strategy on public health, the review team will decide if there's a sufficient data to draw a conclusion if the ethnic difference existed. If the data is not sufficient, BSE cannot be waived. We will require more bridging data, and CDE can provide a consultation meeting. If the ethnic difference existed, BSE could be waived, or it might require a more study for proper dose in Taiwanese population, and it also need a more discussion in NDA review process. Sometimes the ethnic difference might suspected. It usually happen in some special case or some rare disease. It might be conditionally waived. And the strategy on public health, including a warning addition in labeling, post-marketing requirement, and East Asian study, which is ongoing, and submit a full report once it was completed. Okay, we have known that MRCT, multi-regional clinical trials, was the major source as a bridging data. So we conduct several database analyses about multi-regional clinical trials. It's our history scope in Taiwan BSE application. In 2021, these drugs were required to submit BSE. NCE, new biologic, vaccine, blood product, allergen extract belonging to new entity. And the following drugs was not required to submit BSE. Biosimilar, orphan drug, cellular and gene therapy product, and drugs receiving designation of treatment of pediatrics and rare serious disease from TFDA. It's my data collection. In the past 10 years, there were 507 cases in BSE. And I exclude this data based on our scope. So there are 382 cases in my analysis. I was to see if there's a trend and correlation about Asian percent and the BSE result. And there's 351 clinical trials, MRCT, including Asian region. I also want to see if there's a trend. So let's first look at the BSE cases. How's the contribution of multi-regional clinical trial in bridging study? The y-axis is the number of BSE cases. The blue bar is a BSE case. As you can see, since 2016, the BSE case was reduced with time. It probably because biologics accounting for the major in pipeline. And we also mentioned that for drugs with designation of treatment of pediatric and rare serious disease, this drug doesn't need to submit BSE. And the orange bar was the BSE case submits MRCT including Asian region. As you can see, about 60 to 70% of cases submit MRCT as their bridging data. Okay. Let's look at this figure. The y-axis is still the number of BSE or MRCT. The first yellow line is MRCT numbers, including Asian region. The number of MRCT increased with time. And the orange line is about the BSE cases submitting MRCT, including Asian region. The difference between the two lines getting larger with time, which means more than one MRCT submitted in one BSE application in recent years. In this analysis, we also observed several situations. The MRCT of machine medicine was limited in Asia, and there's no arrangement of MRCT in topical medication because of the systemic absorption of topical product was extremely low. So sponsors used now to spend an MRCT in Asia. Okay, then the next part about the multi-regional clinical trials. In our database, most cases were phase 3. 81% were phase 3 trials MRCT, and 13% MRCT were phase 2. 6% were phase 1 MRCT. Then let's further see details about early phase. The green bar is still the phase 1, and yellow was still the phase 2. As you can see, with time, the early phase MRCT was increased in Asian region. Okay, it's an Asian map about the participation of country in MRCT. It's a data from 2017 to 2021. In these 4 years, the total number of MRCT which was conducted in Asian region were 202 clinical trials. And the top 3 countries' participation in the were South Korea, Japan, and Taiwan. This means in these 202 clinical trials, 53% of these clinical trials opened their clinical site in Taiwan. Though the participant in China and Hong Kong was low, but if we see the enrolled subject number, they were quite considerable. And I want to mention again, not all the data, all the countries on this map were considered as East Asia. As I mentioned earlier, the data from East Asia, including South Korea, Japan, Taiwan, China, Hong Kong, was considered better as a bridging data for Taiwan. Okay, how's the waiver rate if Taiwan was involved? In the past 4 years, 13% cases was not waived. And 62% the cases was waived with the Asian region in MRCT. And 25% the case was waived without Asian region in MRCT. This might be they have conduct other trials or local trials in Asia. And let's see this part. And I change it into a bar. You can see there's a higher waiver rate in cases with Taiwan in MRCT. Okay, and then how many Asian percent was sufficient in MRCT? Each point could be presented as one BSE case. I divided this case into 3 groups: waiver, conditionally waiver, and non-waiver, and I ranked them with time. The ratio, the y-axis is a ratio of Asian in selected MRCT. The selected MRCT usually would be a pivotal phase 3 trials with the most Asian in trial. It seems there's no significant trend in this figure, but we still can figure out some tips. Okay. All these figures were waiver case. As you can see, there's still some case was waived with low Asian percent in their MRCT. In 2011 to 2017, there are 40 cases was waived with the Asian lower than 20% in selective MRCT. But there are 75% of the case, actually they submit more data, but more phase 2 or 3 MRCT, or they conduct a local trial in Asia. In recent 4 years, there are more cases submit more Asian data by MRCT or local trial in Asia. And in this group, there are 3 cases was waived with limited data. And the reason was a low incidence rate, sufficient experience review in the central class, and there's one have a ongoing bridging study in Taiwan. Okay, let's look at the conditionally waived case. These were cases was conditionally waived. We know that conditional waiver usually happen in some cases with unmet medical need, so limited Asian data was expected. But how can there's some 2 cases was not waived, even they submit a high Asian percent? The deficiency and requirement was where Asian data didn't cover the maximum dose, and the selected trials was required once they completed. In one case, the safety profile was concerned, so there's a more post-marketing surveillance was required. Okay, then let's look at the last non-waiver case. The 3 case was not waived, but they submit a high Asian percent. Why these 3 cases cannot be waived? Because they have a major deficiency in CCDP, include inconsistent efficacy in Asian population, failed phase 3 trial, and efficacy endpoint of study design was concerned. So that's why they cannot be waived, even they submit sufficient Asian data. Okay, it's my last slide. In conclusion, the availability of ethnic comparison was required in bridging data. It should contain sufficient East Asian and matching Western data, including PK and clinical. For non-single-use drug, multiple dosing PK from East Asian patient was preferred. And there's a trend more than 1 MRCT conducted in Asia in one pipeline, and more PK/PD data in early phase was expected. And also, there's a higher waiver rate if Taiwan was involved. So we encourage to have more MRCT conducting in Taiwan. And thanks to my colleague, thank you for your supporting. And I want to special thanks to my colleague, Hui Chun, Mongshen, and Qi-Yun for their data collection. Without them, I can complete all these analyses. Thanks for your attention.
Thank you, Ms. Wang, for your insightful talk. Thank you very much. Thank you, and we also thank Dr. Ye for moderating the session and all the speakers in this session. Thank you. Now, ladies and gentlemen, we're going to take 20 minutes break. Our next session will begin at 2:50. Thank you, and for those who are joining us online, please feel free to raise your question by clicking the link at the bottom of the YouTube screen, and you can type your question in either Chinese or in English. We will take 20 minutes break. Thank you. We will now begin the last session of the day. We are very honored to have Dr. Ming-Xiao Zhan as our moderator. Please welcome Dr. Zhan onto the stage.
Okay, good afternoon. Welcome back. Please hang in there. And it's my great honor to chair the final session in today's conference. In the next 90 minutes or so, we are pleased to invite 3 speakers, one from regulatory, 2 from industry. They are going to share with us the implementation of bridging study evaluation from different perspectives. Okay, our first speaker is Dr. Jixun Chen. Dr. Chen is a medical doctor graduated from Chang'eng Medical College. He had his professional training as a neurologist at Chang'eng Memorial Hospital. He has joined CDE Taiwan since 2005, and now as the Senior Clinical Section Chief, Division of New Drugs. Today, he will be sharing his expert opinion on the clinical considerations of bridging study evaluation and case studies from regulatory perspective. Dr. Chen Jixun, please.
Thanks for your kind introduction, Dr. Jan. And ladies and gentlemen, in this session, I would like to talk about the clinical considerations of bridging study evaluation and give some examples of ethnic differences. This is my outline, including a brief introduction of bridging study evaluation, some special ethnic difference factors in Taiwan, the principles of clinical evaluation of ethnic difference in bridging study evaluation, and finally showing 3 cases of different scenarios. BSE is a regulatory procedure in Taiwan. It has been implemented since year 2001. The purpose of BSE is to understand whether the efficacy and the safety data from foreign region could be extrapolated to patients in Taiwan. In other words, the major task of BSE is to evaluate ethnic difference and give solution if needed. BSE is part of new drug review. In addition to NDA approval, bridging study must be waived for final licensing. In year 2001, the scope of BSE submission included new chemical entity, fixed dose combination, and a new route of administration. However, currently, the scope was narrowed down to new chemical entity and biologics only in order to facilitate new drug approval. For cases BSE submission not required, ethnic difference will be evaluated in NDA review. It should be noted that assessment of extrapolation of foreign data is nothing to do with the benefit-risk evaluation of foreign data themselves. Comprehensive benefit-risk evaluation will be done in NDA review. That is, if the benefit-risk ratio of the foreign data is unfavorable for approval. Such results will be extrapolated to Taiwan patients if there is no ethnic difference. The most important scientific basis of BSE is ICH E5 guideline. The purpose of this guideline is to provide methodology of how to accept data from foreign region for a new region in order to minimize duplication of clinical data. ICH-E5 provided methods to characterize ethnic sensitivity and introduce the intrinsic-extrinsic factors for evaluation. The most important concept is to provide bridging study strategy for extrapolation of foreign data to new region. The ICH-E5 guideline also described various types of bridging studies for different scenarios. The revised Q&A also indicated that multi-regional clinical trial could be served as a bridging study if a region of interest was included. Taiwan version of ICH-E5 is available. The contents were basically translated from original E5 guideline. The design of MRCT was discussed in ICH-E17 guideline, which could be regarded as extension of ICH-E5. This is the 12-item checklist which has been shown by previous speakers. The checklist was developed by Taiwan regulators according to ICH-E5. ICH-E5 is not the only strategy to accept foreign data. Actually, before year 2001, Taiwan Regulatory Authority required local clinical trial of 40 subjects without evaluation of ethnic sensitivity for all new drugs developed outside Taiwan. Some other regulatory body in Asia also required local clinical trials without adapting ICH-E5. In ICH-E5, it clearly mentioned that if bridging study failed to verify efficacy and the safety in new region, additional confirmatory trial would be necessary. For some cases that Asian phase 3 clinical trial may be more appropriate instead of bridging study. Standalone drug development in East Asian region might be necessary for certain cases in which bridging study are not appropriate. For example, if the target dose should be changed or reduced due to important ethnic difference. In Taiwan, we have some distinct conditions which may affect the BSE. The first one is hepatitis B and C. Hepatitis B and C are chronic diseases which may be reactivated by some immunosuppressants, resulting in clinical exacerbation. The prevalence of hepatitis B carrier in Taiwan is 7.85%, which is higher than that of the United States. So additional risk minimization measures may be required for some immunosuppressions in Taiwan. Another condition is the venothrombotic events after orthopedic surgery of lower limbs. Most of the new anticoagulants seek for the indication of VTE prevention after surgery of lower limbs. However, according to Taiwan observational studies, the incidence of VTE is low after total knee replacement and total hip replacement, and anticoagulant was not routinely given in Taiwan. These results may impact on benefit risk evaluation of new anticoagulants claimed for such indication. Now the tuberculosis. The incidence of tuberculosis is higher in Taiwan as compared to North America, indicating higher prevalence of latent tuberculosis. Some immunosuppressants, such as TNF-alpha inhibitors, may reactivate latent tuberculosis. So latent tuberculosis screening is recommended before initiation of certain immunosuppressants. However, latent TB screening by traditional tuberculin test is not applicable in Taiwan due to universal BCG vaccination since 1951. Therefore, we have a Taiwan risk management plan for latent TB screening using more complex methods. Now I would like to discuss the review principles of BSE from clinical point of view. There are two regulatory procedures for new drug approval in Taiwan: BSE submission and the NDA submission. We integrate the BSE to the NDA review all the time. The material required for BSE submission were included in this slide. The sponsor should provide draft label or clearly describe the claim indication and the recommended pathology. The sponsor should also provide complete clinical data package. This may contain all clinical trials from phase 1 to phase 3 related to the claim indication. The materials should be arranged in CTD format. However, for BSE submission, Module 2 may be adequate, but sometimes the reviewer may request Module 5. Pharmacokinetic data of yeast agents must be included in CCDP. The bridging data package is the data of yeast agents, including PK/PD and the clinical efficacy and the safety. The bridging data package may be obtained from CCDP. In addition, the sponsor should complete the self-evaluation checklist as shown in previous slide. The BSE review team includes PK reviewers, clinical reviewers, and a project manager who is responsible for logistics and communication. This slide shows our review strategy of BSE. Characterization of the ethnic sensitivity of the product will be assessed first through the review of the 12-item checklist. Then the reviewer will compare the PK/PD results between Caucasian and the yeast agents. Comparison of PK/PD profile is essential for justification of the target dose for yeast agents. Finally, we will compare the clinical efficacy and the safety results. There are several forms of clinical efficacy and the safety data of yeast agent. Bridging study is the traditional one. Yeast agent phase 2 dose response results are very useful to determine the similarity between two regions. Subgroup analysis of East Asian subjects of MRCT is also acceptable if the sample size of East Asian is adequate. But there must be no important ethnic difference from PK/PD comparison to justify that same target dose can be used for all regions. A phase 3 randomized control trial conducted in East Asian population will provide direct evidence, but it's only required in certain cases. As I mentioned in previous slide, bridging study waiver and the favorable benefit-risk ratio of NDA is essential for final NDA approval. So here are the impacts of BSE resolutions on final NDA assessment. If the submitted materials of BSE are inadequate for review, the bridging study will not be waived and the NDA will not be approved. If the BSE shows no or little ethnic difference, we will adopt the following data and there will be no label modification or regulatory actions. If there is substantial ethnic difference but no clinical significance, there will be no label modification or regulatory action either. But if there is clinically significant ethnic difference, we may require label modification of indication, pathology, or special warnings. Sometimes risk minimization measures or spatial pharmacovigilance is required. Finally, if the bridging data cannot extrapolate foreign data to Taiwan patients, further East Asian data will be required and the NDA will not be approved. Usually, bridging study evaluation is assessed case by case, considering various factors such as clinical importance of ethnic difference, unmet medical needs, availability of flexible doses, and the feasibility of risk management plan, etc. Finally, I will give 3 cases of ethnic differences with different scenario for each one. The first case is prasugrel. Prasugrel is a new generation thienopyridine antiplatelet agent, and it is a prodrug. The active metabolite is R13A727. The indication is acute coronary syndrome with percutaneous intervention. The global pivotal study is Triton-TIMI-38, which enrolled mainly Caucasian subjects. Based on this study, the approved pathology is 60 mg loading dose and a 10 mg per day maintenance dose in the United States and Europe. However, the approved pathology in Japan and Taiwan is 20 mg loading dose and 3.75 mg maintenance dose, almost 1/3 of a Caucasian dose. So let's look at the ethnic difference of PK/PD. The PK profile of active metabolite show that the AUC is 1.4 times higher in Japanese as compared to Caucasians. And the C maximum is 1.2 times higher. However, the most important ethnic difference is pharmacodynamics. In this figure, IPA is used as PD biomarker. The hollow circle represents Caucasian. Okay. The other symbols represents East Asians from 3 countries. So this symbol represents East Asian. So we can find substantial higher PD effect in East Asian after Caucasian dose. Higher PD may indicate higher bleeding risk. Therefore, target doses were proposed. Lower target doses were proposed for East Asians. In this figure, PRU is used as PD biomarker. This is PRU. The lower, the more effective. The hollow square represents standard dose of clopidogrel. So this is the curve of clopidogrel, the comparator in the pivotal study. The solid circle and the square represents 2 doses of prasugrel. So these 2 curves represents 2 lower doses of prasugrel. It could be noted that after 4 weeks, the PD effects are still apparently higher in low doses groups of prasugrel as compared to those of clopidogrel. So these 2 curves represents lower doses of prasugrel, and the lower, the more effective. So apparently, 20 milligram loading dose with 5 milligram maintenance dose may be adequate for, may be appropriate for East Asian. So 20 milligram loading dose and the 3.75 maintenance dose was selected as target doses in Japanese phase 3 study PROSFIT ACS. The study design is similar to the global trial, Triton-TIMI 38. The results of Japanese study show that the efficacy is comparable with Caucasian trial. Even the survival curve is very similar. Most of the event occurs during the first 24 hours. The pattern is very similar to the global clinical trial. The hazard ratio of MACE is the primary endpoint of the 2 pivotal study. The hazard ratio of MACE is 0.77 for Japanese and 0.81 for global trial. How about the bleeding risk? The bleeding risk is apparently a little bit higher for Japanese. So selection of those is very important. We must keep the efficacy, but lower the risk of major bleeding. Okay. So this case indicates that if ethnic differences is important and critical, then the target dose should be modified, and the development strategy may not follow ICH E5. The second case is Agranox. Agranox is a fixed dose combination of 20 milligram aspirin and 200 milligram dipyridamole. The indication is to reduce the risk of stroke in patients with previous TIA or thrombotic stroke. The recommended pathology is 1 capsule twice daily. Taiwan regulators noticed that the rate of discontinuation due to headache in Asians was twice as high as in Caucasians. Therefore, a bridging study was required. So this is the study design of bridging study. It is a randomized control trial. Compare the risk of a headache for the 3 different dose regimens. The placebo group received placebo from day 1 to day 28. The dose titration group received placebo from day 1 to day 4, then 1 capsule of Agranox before sleep from day 5 to day 14. Then 1 capsule twice daily for the remaining 2 weeks. The regular dose group received Agranox 1 capsule twice daily throughout the 4-week study period. The primary endpoint is the prevalence and intensity of headache and the patient's tolerance. Now here are the results. The study results show that the risk of headache and the discontinuation due to headache was higher without dose titration. The risk of headache was comparable between dose titration and the placebo. So you can see that the rate of headache is 15.2% and is very similar to the titration group, but apparently higher in regular dose group. The most important parameter is rate of discontinuation due to headache, because this result may be related to the compliance, dosing compliance. So you can see the rate of discontinuation due to headache was comparable between placebo and the titration group, but apparently higher in regular dose group. So accordingly, we recommended dose titration for patients with headache during initiation. And we found that similar recommendation also appeared in US label. But someone may ask whether such regulatory action may compromise the efficacy. Yes, probably, because patient needs dose titration for several days, even 2 weeks. So what if the stroke happened during these 2 weeks? Okay, that's the problem. This is the problem where the bridging study didn't explore. But in clinical practice, it's very simple, very easy to resolve this problem. You know the regular dose for aspirin is 100 milligram per day. For Taiwanese patients, we know that the lowest effective dose to prevent ischemic stroke is 50 milligram per day. So during the titration group, the physician may add 100 milligram aspirin to the patients. So that's enough for regular prevention. So even though this bridging study cannot prove the efficacy for dose titration, but in clinical practice, I am a neurologist, I will prescribe additional 1 tablet of 100 milligram aspirin to the patients. That's what will resolve the problem of risk of recurrent stroke during the titration period. Okay, the last case is Balversa. It's a relative new drug. The active ingredient of Bavarsar is erdafitinib, which is an FGFR receptor inhibitor. The indication is locally advanced or metastatic urothelial carcinoma. The pathology is 8 to 9 milligram daily by oral administration. Dose reduction according to phosphate level and adverse reactions are recommended. The pivotal study is BLC 2001, which is an uncontrolled trial demonstrating response rate as primary endpoint. This case was reviewed by accelerated approval pathway. Therefore, a confirmatory trial is required for regular approval in the future. In bridging study evaluation, there is uncertainty of ethnic difference. The exposure of erdafitinib is higher in Japanese as compared to non-Japanese. There is few East Asian subjects for subgroup analysis for efficacy because the pivotal study is the phase one trial. So we don't have enough East Asian subjects for meaningful subgroup analysis. However, totally there are 59 Asian subjects pooled from data sets of various other cancer trials. From this data set, we found that there is apparently higher risk of hyperphosphatemia and the liver enzyme elevation for East Asians. However, we didn't request additional East Asian data before accelerated approval. The reasons are, first, the indication is of unmet medical need. Second, even if high risk of East Asians do exist, dose reduction according to adverse reaction is applicable, according to the label. Okay. Finally, the apparent higher rate of hyperphosphatemia and the liver enzymes elevation could be monitored in clinical practice. We can prescribe a blood test for electrolyte and the liver enzyme, maybe weekly or every 2 weeks. But further East Asian data are required in post-marketing setting. So maybe the confirmatory trial, the phase 3 confirmatory trial may provide additional East Asian data. The most important factor is that the indication is of unmet medical need. It's for last line of urothelial carcinoma. So we granted accelerated approval without a request for additional East Asian data. So, in conclusion, bridging study evaluation is based on the principle of ICH-E5. This guideline provides methods of ethnic sensitivity, characterization, and the bridging study strategy to accept foreign data. Our review strategy of BSE, including assessment of intrinsic and extrinsic factors and comparison of PK/PD, efficacy, and safety between East Asian and the Caucasian. There are various forms of East Asian clinical data. Bridging study is the traditional one, as described in ICH-E5. As a clinical reviewer, I think a phase 2 dose response study is very useful because the similarity of the dose response curve between two regions may give good evidence to accept foreign data. MRCT could be used as bridging study if there is no important ethnic difference from PK/PD comparison. MRCT is more efficient for drug development, enabling simultaneous NDA submission globally. In some cases where ethnic difference is substantial and critical, or when target dose should be changed, East Asian phase 3 study may be required. So, thanks for your attention. I would like to answer any question in panel discussion.
Dr. Chen, thank you for your presentation. I think the bottom line is that the Asian data is highly valuable in terms of bridging study evaluation. And without adequate data for assessment, I think the evaluation of ethnic factor will be difficult and challenging. Okay, we are a little bit behind our schedule, so let's move on to our next presentation. The next speaker is Ms. Louise Watt, also a special guest from Singapore. Ms. Louise Watt is Regional Senior RA Manager in Novartis. She has more than 15 years of working experience with several international pharmaceutical companies. She also has extensive experience working with regulatory health authority in Asia Pacific to support development and resolution of complex regulatory issues. Her talk is about the implementation and consideration of ICH E5 in Asia region from global pharmaceutical industry perspective. And please welcome Louise Watt.
Good afternoon, distinguished guests, members of the organizing committee, all speakers and participants. I am privileged and honored to be here as a speaker and industry representative from IRPMA. I will share the global pharmaceutical industry experience and perspectives on the implementation and considerations of ICH E5 in Asian regions. Before I start, there are a few disclaimers that I would like to make. The opinions expressed in this presentation and on the following slides are solely those of mine and not necessarily those of Novartis. Novartis does not guarantee the accuracy or reliability of the information provided in the slides. The key objectives of my presentation today is to share the experience of MRCT under E5 over the past decade in Taiwan and Asian regions from a global pharmaceutical industry perspective, and also to provide the latest updates and perspectives of drug development approach in global pharmaceutical companies. The ICH E5 guideline was introduced in February 1998 and subsequently implemented by the regulatory authorities of US, EU, Japan, and other health authorities in Asian regions. The main objectives of E5 are to prevent unnecessary and redundant repetition of clinical studies in order to bring medicines to market in different regions, while adequately evaluating the impact of ethnic factors on the efficacy and safety of drugs. E5 also outlines the use of bridging studies, if necessary, to allow extrapolation of data and development strategies with regards to ethnic factor influence on the safety, efficacy, and dosage of new medicines. E5 guidelines also provide clearer definition of what constitutes an ethnic factor. Ethnic differences are divided into intrinsic and extrinsic influences. In 2001, the Bridging Study Evaluation Review Process, BSE, based on the ICH E5 guideline, was introduced in Taiwan. The scope of the BSE review included new chemical entities and new biologics. In September 2021, an exception was introduced for new drugs with available clinical data in Taiwanese patients alone and available PK data on ethnic groups in East Asia. This update could accelerate the development of domestically developed new drugs and bridge to new drug development approach under ICH E17 guidelines. In filing the BSE in Taiwan, the complete clinical data package, East Asian data, as well as Taiwanese checklist is required in the submission package. This slide shows the checklist for BSE submission in Taiwan. This checklist is based on ICH E5 Appendix D, and what is highlighted here is that the ethnic sensitivity information on pharmacokinetics, pharmacodynamics, dose finding, safety efficacy data in Taiwanese and or East Asians, and its comparison with Caucasian patients are required information for BSE. This information is usually prepared in the format of an ethnic insensitivity report in Novartis. In a CDE publication dated 2016, CDE analyzed the data from 2004 to 2015 to find the correlation between East Asian Taiwan data and BSE results. As shown in the bar chart, the number of non-waiver cases decreased steadily over the years. CDE analyzed the reasons for these non-waiver cases. Majority of the cases, which is about 68% of the cases, did not have clinical data in East Asian population, or there was insufficient sample size of East Asian patients. This emphasizes the importance of East Asian data to achieve bridging study waiver from the CDE. I would like now to share the Novartis BSE cases. There are 15 cases evaluated between 2015 to 2021. Bridging study waiver was achieved for 14 cases, and 1 case was rejected because of insufficient safety and efficacy data in Asian population. 57% of the bridging study waiver cases included sufficient Taiwan, East Asian data, while 43% of the cases included sufficient Asian data, however, with no Taiwanese data. It is interesting to note that these cases that did not have Taiwanese data are all oncology therapeutic products. These cases reflect the importance of East Asian data to achieve bridging study waiver from the CDE. When we look deeper into 3 NDA bridging study waiver cases to better understand the Taiwanese and East Asian sample size for bridging study waiver, we find that the more Taiwanese patients, the higher the chance of success for bridging study waivers. Brand A had 10% of Asian patients in global phase 3 studies, and Brand B had 23% of Asian patients in global phase 3 studies. Brand C is an in-licensed product. It had a very low percentage of Asian patients in global pivotal studies, only 1.4%. However, Japanese PK data was available. We were able to secure the bridging study waiver for Brand C. However, we have to do a post-approval commitment. There are several considerations under E5 in Asian region that impacts development and regulatory strategy. Participation in global drug development at early stage, be it phase 1, phase 2, and in later stage phase 3 is needed to secure bridging data. Ethnic insensitivity report or subgroup analysis report are required to allow extrapolation of foreign clinical data to Asian region. This is provided there is no major difference on PK, similar dose response relationship between Asian, East Asian, and foreign population, and no major difference in safety. For a Taiwan marketing authorization application, the more Taiwanese patients, the higher the success rate of bridging study waiver. Moreover, East Asian data from Malaysia, Thailand, Singapore, and Vietnam is accepted. Japan, Korea, and China require Japanese, Korean, and mainland Chinese patients, respectively, for their marketing authorization approval. As we gain more experience in developing bridging studies under E5, we also face challenges. Due to difference of study condition, for example, the timing of the study of the bridging studies is different from the investigational organization, etc., it will be difficult to compare the efficacy and safety between bridging study and reference study. Inter-study condition difference may also lead to a certain bias. In order to eliminate bias to study result and be able to better compare across regions, multi-regional clinical studies is preferred. Indeed, the new drug development approach in global pharmaceutical companies is evolving. Currently, it is more common to conduct phase 3 multi-regional clinical trials under ICH E17 with an appropriate number of subjects from Asian regions. Asian region will include Japan, China, Korea, Taiwan, and rest of Asia. Asian PK data is collected from PK studies conducted in Japan and China, in addition to these countries joining phase 3 multi-regional clinical trials, or such data may be collected from global PK studies. Regional bridging studies or local standalone studies are conducted in certain cases, for example, when the product is in license with no available Asian data. However, this is not common. This slide and the next two slides show a trend of increasing number of multi-regional clinical trials conducted in Japan, Korea, and Taiwan. This reflects the development approach of new drugs in recent times, as I've described in the previous slide. With multi-regional clinical trial E17 approach, the sample size is a hot topic in the design and planning of such studies. Japan issued a guidance in 2007, Basic Principles on Global Clinical Trials, in which they described the PMDA Method 1 and Method 2 statistical approaches to determine the sample size of Japanese patients in multi-regional clinical trials. China NMDA recommends proportional allocation and equal allocation approaches mentioned by E17. Practically speaking, PMDA Method 1 and Method 2 may be used. The general principle is that specific sample size should be decided on a case-by-case basis, taking into account the general considerations. Korea bridging cases have been based on ICH E5, which requires Korean patients. To fulfill bridging requirements in general, 10 to 15% of Korean patients should be included in global pivotal study, or 27% Koreans included in regional studies, or 100 Korean patients. Taiwan, in principle, the more Taiwanese patients, the higher the success rate of bridging study waiver. Moreover, East Asian data is accepted. Looking ahead, the global pharma industry will continue to gain experience on the understanding and the interpretation and implementation of E17 guidelines in the planning and design of multi-regional clinical studies. In conclusion, I would like to share some reflections and recommendations. Global pharmaceutical companies are frequently using multi-regional clinical studies in global drug development to promote simultaneous development and patient access to innovative drugs in Asian region. The ICH E17 guideline was developed to increase MRCT data acceptance by multiple regulatory authorities. The paradigm shift to MRCT under E17 may warrant a waiver of a separate BSc submission in Taiwan. It is important that global pharmaceutical companies work closely with regulatory authorities to establish an understanding on design and planning of MRCTs in specific development programs. Besides sample size, choice of comparators, inclusion and exclusion criteria, and pooling strategies are also important topics to be addressed. Exchanges of experiences and consultative engagement between industry and regulatory authorities will increase awareness of the issues and challenges of implementation and overall yield positive impact. On this note, I end my presentation. Thank you very much for your kind attention.
Okay, Louis, thank you for your presentation. Okay.
Thank you very much.
Yeah, I have a question that, do you have the camera for your device? Because we could not see your face. Okay, that's good. Yeah, lovely to see your face, yeah, although it's virtual. Okay, thank you. Okay, we are a little bit okay, but that's good. Again, from Novartis sharing, we could see that East Asian data is so critical to get a BSE waiver. However, even though Taiwan adopts a more flexible approach, but although the Taiwanese data is not mandatory, but it is highly expected from review perspective. So I would say Taiwanese data is definitely a bonus. Okay, and at last, personally, I'm very positive to see the paradigm shift to MRCD under E70. Okay, let's move on to the third presentation. The next speaker is Dr. Jian Du-Xian. Dr. Jian is the founder and or chairman of multiple biotech companies in Taiwan and US. He was also the key inventor of several global patents of new chemical entity and is outstanding in academic achievements. He's going to talk about the implementation and consideration of ICH E5 in Asia regions from Taiwan research-based industry perspective. Let's welcome Dr. Jian Du-Xian.
Thank you, Dr. Chang's nice introduction. I'm not running for the mayor of Taipei City, so basically I'm going to talk about the regulatory and the E5 section. But before I start to talk, I want to thank you staying for the last talk, which I'm giving very soon. And second, I want to thank you for the conference committee to invite me. Perhaps the only one from the local pharmaceutical company. So I'm going to talk about presenting the pretty much the Taiwan local biotech and drug company. So on behalf of the TRPMA, I will talk about our perspective on the ICH E5 in terms of the filing, also the approval. Let me start to introduce a little bit about TRPMA. Should I stay in here? Yeah, okay. Okay, TRPMA established about 2012, and right now it's about the 52 member company. I think just yesterday we increased about 65 because the medical device company also start to join the TRPMA organizations. This is probably a very unique research-based pharmaceutical cluster in Taiwan. It means a lot of drugs, the new chemical entities or the 505(b)(2) drugs, all focus on these companies. So the members, they are developing new drugs, regenerative medicines, medical technologies, also digital health. That's pretty much focus the government demand right now. And this is a very unique platform for government, academia, also for the industry, the collaboration. So we can talk about lots of the outside regulatory or government policies, issues under this organization. Most drug company, member companies have a motivate innovation. many on the NCE, also new biological entities. They also have a strong alliance with an international partner. This is basically the TRPMA. TRPMA members came from, I would say, mainly from 3 parties or 3 groups. On the left side is the new drugs. So this is a representative company in Taiwan, only focus on new drugs. In the middle part, about 20 companies focus on regenerative medicines. On the right side, you will see that the innovative medical device. I think on the right side, innovative medical device, the member will increase in the next 5 years. What we did is for the supporting the E5, ICH E5 for the BSE waiver, the 25 member company, they're about the 12 companies, about 44% focus on only small molecules, mean chemicals. On the left side, on bottom part, that's a focus on 505(b)(2), meaning the drugs already, molecule already approved in Taiwan or other drug company in the other global area, which we modify the formulations, or we change indications, or some of the company, they are doing a combination of with other drugs. They all belong to the 505(b)(2). 6 companies doing that is about 20% of the overall members. On the left part side of biological, probably 8 companies doing that about the 30%. On the right side, you will see current status of drug development for the member company, all conduct in various countries at the various stages from the phase 1 all the way to NDA and marketing. What I want to point out to show you is that the first 2 countries, Taiwan, US, you will see that the majority of these cases actually fall in Taiwan and US, from the phase 1 to phase 3. You will see that many studies conduct in Taiwan and US parallelly in the phase 2 study, but actually many companies start on the phase 1. It meaning that those companies, we want to determine whether there's ethnic differences in the very beginning from the small molecule. So we submit, normally we submit the IND to US. Then once approval, we come back to submit to TFDA, IND in Taiwan, TFDA. So we conduct a study parallelly. For the small molecule, we really want to understand whether the metabolism phase or the toxicology response is between the Caucasian and Asian, the same or difference. I think that there is very important strategy for the most Taiwan companies doing right now. One of a country, 2 countries we are very like to explore more, but there's a lot of regulatory hurdle or commercial hurdle, including the current conflict situation, I would say that. So including China here. You will see that's actually fall in the number 4 here, only 14 cases being submitted to China. Japan, only 3 cases. The reason probably already reported this morning, many difficulty to enter those countries, but we're still working on that direction. So what we did is in order to support the BSE filing submission current status, the TRPMA sent out a survey. We sent out the questionnaires to the member companies, then ask them the status of the filing BSE in terms of Taiwan, China, Japan, and Korea. So we're not talking about the US, we're talking about Asia, these 4 countries. Then we also want ask the TRPMA member to identify what type of a product they submit, whether there's an NCE or new biological entities, or this is 505(b)(2), or something else from the drug-derived derived from the genetic engineered vaccine. Okay, so we want to identify what product actually they submit for the BSE to these countries. The questions we are asking is very different from regulators asking or from the government. We asking more in terms of the interaction. Okay, this we are tracking back to only 10 years because the TRPMA only established about 12 years ago. We asking about the interaction, about the feeding. So they're very different from what you are talking this morning. We want to know what's the interaction with the Taiwan TFDA and CD. We put them together as a big family. Okay, so we want to know during our submission, what's it look like? During the review process, what does it look like? It's actually is a positive that encourages a lot of drug companies in Taiwan. They will first consider to submit their product to that specific regions because there's a higher chance to get approval. Then the third question we ask, what's the interaction with the foreign regulatory agency, including China, Japan, Korea? Again, during the submission and review process. The fourth question is, actually we are more concerned in terms of the drug industry or the sponsor. Once we submit the BSE and get approval, then essentially product get launched and market in that specific country or region. What those product will do? Are they really come out the efficacy and safety as what we expected before we apply for the waiver? We really like to tracking what's it look like, and most company will pretty much want to follow. It's the BSE. It's a regulatory pass. It's a very speeding, facilitate the process, but the outcome is to come out as what we expect or not. So I'll show you the result. We send out a questionnaire, then come back about 10, including 6. They have said they have a BSE submission in Taiwan. This is all from the local drug company. 5 are NCEs. 1 is a non-drug of a new formulation or new indication. This is a 505(b)(2) typical. Maybe different country, different name, but we know what are we talking about. Basically, the non-drug molecular. 1 product derived from the genetic the engineering. Okay. Now, you can see the tables here. The TRPMA member company did not submit the BSE until 2015. Probably at that time, most companies start still struggling with the very exploratory research or just still in the preclinical stage. But 2015, the member company start to submit. You can see the first 2-3 years all in Taiwan. Okay. So some to 2021, total 11 submit to Taiwan. We got 10 approved. Very high rate, very high rate. Okay. China, we start submit to 2018. We submit 3 and only 1 get approved. Japan submit 1. I think so right now still under evaluation. It take a long time, as you can expect it. Lots of detailed questions. Korea, 2 submission, 2 approval. Okay. So this table show you what's the current status of a Taiwan drug company submitting the BSE. Now, this is the interesting part. What's the interaction between the local company and the local regulatory agency? Let's start from TFDA and the CDE. When company indicates very satisfied, okay, 4 show satisfied, 1 is natural, but I take an average 4, satisfied, means good. You know, we don't give a very satisfied, but to the agency, I think the 4 is already on the top. But I want to see the comment. I'm going to show you the comment. This is very important. Almost all drug companies indicate this a good interaction with the TFDA and CDE. Friendly, smoothly and clear. This is a lot of credit. It's not easy to sponsor a drug company to say that. But I think these are two points are very important, even prior to the submission. The CDE offer consultations. So we got a chance to meet with a CDE reviewer, maybe get some instruction. This is a meeting with a CDE. It was very productive and very helpful. Keep in mind, most of the biotech companies in Taiwan are still very young. They don't have a lot of experience. They don't have a lot of talent from the foreign country or big pharma. But they're seeking a lot of help from the CDE. CDE often require a lot of support for during the submission, even till the granted. Okay, this is a very important comment. And last one, I think is the most important one. CDE provide clear regulatory instruction, meaning what you need to prepare. I don't think just the one table you show several times, that's the whole list. You understand, but most people don't understand what it is. For CDE, the reviewer and consultants, they will give a very clear instruction what exactly needed, and also show the procedure, how to prepare and then submit a BSc. I think this is came out with a 90% of approval rate. Actually, it's not from company, actually from you. Without CDE's help, it's impossible to achieve that. Now we compare to the other three countries, China, Japan, and Korea. China, we got satisfied. Two companies show that one show unsatisfied. This is usually we don't put that the very strong term like unsatisfied to the regulatory agency. Japan satisfies one company, and Korea satisfied, very satisfied. The comment is also important. In China, the 5.1 class new drug, this is a very new drug. The one company report that the drug already approved and launched in Taiwan. NMPA still requests sponsor to conduct at least one phase 3 clinical trial in China. Okay. It's meaning even drugs were already approved by Taiwan, were already launched. In China, it's not necessary. They got granted. They still request for the standard one phase 3 pivotal trial. Sometimes the interaction with NMPA reviewers can be very confused and time-consuming process. I think this is a very lack of the instruction, what exactly you need. Difficult just to say you don't have this, you don't have that, but have to say what exactly. I think that's the lacking the help from the China regulatory agency. Korea, otherwise, most of the product is a 505B2 or called IMD in Korea. BSE is granted. Interaction with the Korea agency is very smooth and helpful. Okay, this tells us the Taiwan drug company is moving first. Will apply for BSE in Taiwan. Then next probably Korea. The third probably China or Japan. I thought we very much like to file the BSE in China. Now the important one thing is in case the BSA waiver we granted, then product finally reach market, then what's the product will behave as what we expected before we submitted. So current survey is showing that the post-marketing assessment is generally established, but more focus on safety. I can understand that the safety is more concern and more focus from the regulatory agency. The efficacy, they will basically compete with all the other drugs on the market. So the focus more on the safety side, less in the monitoring efficacy responses. This is what's the current status. But fortunately, National Health Insurance Administration initiated the called STR, Health Technology Reassessment on some new drugs. I think this is a focus on both sides, efficacy and safety. Okay. So I want to summarize basically from the local aspect from the biotech company or drug company. we still gain what's a BSE really meaning. Probably just less than 7, 8 years. I believe there's more will come because a lot of drugs conduct all the clinical trial in foreign country. They always have a multiple study, also small part in Taiwan. That will be the strategy. So what the current thinking is, can we conduct a study, combine Taiwan, Japan, US, something like that. So it becomes a 3 or 4 multiple national clinical trial. But Taiwan will be on the part of overall. So that's why we will apply for the BSE in the future. That's what the current thinking from the drug company. Okay. So let me end here. Thank you, everybody, for the attention.
Thank you, Dr. Chen, for your insightful talk. Thank you very much. And we also thank Dr. Zhang for moderating the session and all the speakers in this session. Thank you. Ladies and gentlemen, next we're going to have 40 minutes panel discussion. And now we'll have our staff to take a few seconds to arrange the setting on the stage. 我们现在有请工作人员花一点时间来帮我们设置一下台上的 等一下要进行综合座谈的位置。So it is our great pleasure to have Dr. Li Juanxu and Deputy Executive Director of Center for Drug Evaluation and Dr. Yizhen Sun, Senior Executive Officer of Center for Drug Evaluation as our moderators. And now may I please invite Dr. Xu and Dr. Sun as well as all the speakers in this session, Mr. Jiren Yang, Ms. Yiling Wang, and Dr. Jixun Chen, and Dr. Du Xianjian to come up to the stage for panel discussion. And Ms. Louise Watt will also join the discussion via video conferencing online. So later on, when the floor is open to the audience, please raise your hand and our staff will hand you the microphone. And please state your name and your organization before raising your question. So now, Dr. Xu and Dr. Sun, the floor is now yours.
Good afternoon, everybody. Welcome to the panel. And thank all the speakers from CDE and the industry for joining us in this section. This afternoon, Ms. Yang first talked about current regulation and the review process of bridging study evaluation in Taiwan, followed by giving us statistics and common deficiencies. Then Ms. Wang and Dr. Chen presented a regulatory perspective from PKPD and the clinical point of view. In the meanwhile, both presented us in-depth case studies. Afterwards, Ms. Watt from industry representing global pharmaceutical industry mentioned challenges of bridging strategy, new drug development approach, and MRCT trend in Asian region. Dr. Jian, also from industry representing TRPMA, provided the survey results and overall feedbacks among 27 members in Taiwan. Today's talk are quite rich in content. I personally benefit from all the speeches. The audience may have further questions. Now we have about 40 minutes left for this section. So please feel free to raise your hand and or write down your questions online.
Okay. Ladies and gentlemen, all the distinguished guests, I think we can open the questions to the floor now.
Okay.
Dr. Jian.
Okay,
Yeah,
I'm
please.
Minxiao Chen from CD Taiwan. I have one comment and one question. One comment is for Dr. Jian Duxian. And thanks for your presentation and your encouragement to Taiwan FDA and CD in terms of your interaction experience from industry. And I would say communication is considered very important in any way, especially between the sponsor and the authority. And the efficient and clear communication is absolutely very helpful, not only for the submission, but also for the review. That's my comments. And my question to Louis is, it's more like asking your comments. As we mentioned today regarding BSE evaluation, using East Asian data, Taiwan accepts the data from other Asian countries, and local trials are not necessarily required. But we still look forward more industry-sponsored clinical trials conducted in Taiwan. And today we can see some growth in the past 10 years, but honestly speaking, we do expect more. So I would like to ask your opinion from global perspective. Do you have any recommendation for an Asian country like Taiwan, not so big size, but we are willing to attract more global clinical trials? Thank you.
Yeah, Ms. Wang.
Thank you for the question. Can you hear me? Okay.
Yeah.
Yes,
we can hear you.
Yeah, thank you. Thank you so much for the question. From at least from the pharma industry point of view, Taiwan, globally speaking, yes, it may not be a big market, but in Asia Pacific, it is a substantially big market. So in terms of, and we know that the quality of the clinical studies, operation and the conduct of the study is is of high standards in Taiwan. So in terms of advocating for Taiwan to participate in global clinical studies, I think personally in Novartis, we've had many successes. We've managed to able to provide that voice to the global team to include Taiwan because there are so many good precedents of study and the data delivered is excellent, the study quality. So, but sometimes the challenge from the global pharma industry is also with regards to the sample size. Because with these multi-regional clinical studies, we have to balance the regulatory requirements in different regions, different countries, versus the overall trend. So yes, indeed, we do have to continuously advocate also from the Asian perspective, because personally, I really believe that generating Asian data is very valuable in us trying to understand the new therapy, especially in where we want to promote simultaneous development. So this message is definitely continuously on our mind as we participate in all those global development programs.
Okay, I think Dr. Jan's question is encourage like the global company, yeah, we can attract more clinical trials in Taiwan, because we see the in Ms. Wang's slide, the Korea and mainland China, the local trial or the MRCT. Included the Korean or mainland Chinese are required. So it seems it's difficult at this situation that for Taiwanese clinical trial can have a more opportunity in this stage. So so we want, yeah, maybe in today's presentation, we see many presenters shows that if Taiwanese population, Taiwanese people enrolled in the MRCT, then the waiver rate is higher. So I think this is what the Dr. James want to deliver the burden for this question. Yeah, I don't know if there's any questions on the floor further questions?
We actually have received 4 questions from the online participants. 3 for
Okay.
Dr. Chen and 1 for Ms. Wang. Should I just read them through one by one, or just?
I think maybe you read the first one.
Okay.
Yeah, first.
Okay, so the
And
first
then we
question.
have the speaker answer the first question.
Okay, so the first question is for Dr. 季勋成。这个问题是新成分单方药品 X 成分 A 以及含该新成分的复方药品 Y 成分 A 加上已经核准的成分 B， 如果 XY 同时申请 NDA， 那请教 Y 药品是不是可以引用 X 药品的 BSE 结果，无需再额外执行 Y 药品的衔接性试验。So the English translation of this question is that drug X is a drug with a single new active ingredient A, and drug Y is a compound drug with the new ingredient A and the ingredient B, which has been approved. So if drug X and drug Y apply for NDA at the same time, can drug Y use the BSE citation of drug X without executing additional bridging study?
Okay. Dr. Chen, please. And this question is come from the Daiichi Sankyo Regulatory Affairs Manager.
Hello? Okay. This question is a little bit complicated, so I make a description, brief description of this question. So now we have, can the camera focus on this paper? Okay. So now we have component A. Component A is a new chemical entity. And the component B is an active ingredient which was already approved. And now we have 2 products. Product X contains component A as a single active ingredient. So it's a new chemical entity product. And we also have another product called product Y. It is a fixed dose combination of component A and component B. But component A is a new chemical entity. So although it's a fixed dose combination, but we will regard this fixed dose combination as a new chemical entity. So if you want to submit these 2 products, product X and Y simultaneously, either NDA or BSE, for NDA, then although for current regulation, fixed dose combination is a bridging studies evaluation submission is not required for fixed dose combination. But since product Y contains a new chemical entity, so we regard product Y as a new chemical entity. So the sponsor should also submit BSE for product Y. For product X, it's very simple. You have to prepare all the CCTP, bridging data package, draft label, and the self-evaluation checklist, and the report to evaluate yourself for the ethnic difference. But for product Y, you have component A, so you may use the bridging data package or complete clinical data package of product X. But there is another question. You have to discuss the interaction between A and B. What happened if component A and B used simultaneously? For most of the fixed dose combination, both of the components was approved in this country. So we have a long period or we have clinical experience for each component for a long time. But for product Y, it's a new chemical entity. So instead of, in addition to the discussion of ethnic difference of component A, you have to discuss the possible ethnic difference of A plus B used together, because we don't have any experience, such clinical experience. So that's my my answer. So did I answer your question?
Thank you, Dr. Chen. Very efficient illustration to show this complicated question. Yeah, if the answer you want to ask further question, you can still type your question
Can
online.
I comment on that?
Sure, sure.
Businessly, this is a very risky approach. Not just for a clinical trial design, also, I think Ms. Wang will completely reject it. You have a lot of drug-drug interaction you have to address, and not just metabolism-wise, also efficacious synergy or antagonist you all have to address. The problem is, once you get a clinical result, you don't know what is what. You don't know which is which. Then once, if you show Ms. Wang, they will show you the whole bunch of list. If you all want to conduct those studies, I don't think there is sufficient budget to do that, especially in Taiwan. It's very difficult. This is my comment.
Ms. Wang, do you want to add something?
And for this issue, I think previous has mentioned that the drug-drug interaction between ingredient A and B was concerned. And especially sometimes if the drug B is a booster to increase the drug A, the plasma concentration of drug A in your blood. So the experience PK data from product X might be not sufficient to extrapolate to the clinical experience and PK data of product Y. Yes. So from PK perspective, in addition to the drug-drug interaction, and the plasma concentration, all the PK information and PD information provided, we will specifically evaluate if your previous information could fulfill the bridging data we need in product Y. If so, then the previous bridging data information was acceptable. If not, then more bridging data for product Y will be required. Thanks.
Thank you, Dr. Jian and Ms. Wang, for some the comments for the PK consideration.
So,
and
I would like to add a few more comments.
Please.
Okay.
Yeah.
So, if you want to develop a fixed dose combination, from this illustration, the dose is very important. Please, if you want to develop fixed dose combination, the A component of product, the pathology of A component of product Y and the component A, I recommend the pathology should be the same as product X. So don't use a higher dose for product Y, especially for component A and B itself. If the B was approved in Taiwan for maximum dose of 50 grams per day, then I recommend your pathology of the fixed dose combination is also for component B is also 50 grams per day, and don't change the dose. Okay, So if you change the dose, this case will be more complicated. Okay.
I don't know if there's any further comments for this question. I think we have a last, we provide many comments. I think this is a consultation. We need to take the charge.
Yeah. So I strongly suggest that, let me I forget who asked the question. I strongly suggest the consultation to CDE. We will give you a comprehensive response. Okay.
Yes, this is just principle. Yeah, I'm kidding. If you want to learn more details, the development strategy, please welcome to our consultation program. Okay.
Yeah, I have a question for Ms. Watt. And in your presentation, we share with us 3 Novartis NDA cases, and the 3 cases already got the BSE waiver in Taiwan. And would you please to share with us the regulatory condition of brand C in other Asian countries like China, Korea, and Japan? Thank you.
Okay, so for brand C, it's an in-licensed product. In Japan, I believe is under is going through the NDA process because our company in Japan needed to do a local study. In China, I believe is also under development, separate strategy, and the reason is quite complicated because of the global agreement with another company. As I said, this is an in-licensed product. There are different arrangements in different countries within Asia. So that's why it's quite complicated for brand C.
Okay, thank you, Ms. Hua. Maybe we have we have a question from the floor. Dr. Ye.
Yes. I thank you for the presentation. And just prior discussion, the consultation is very important. And the Manager Yang has mentioned in your slides the consultation pathway. Can you give more information if any sponsor wants to submit or apply this, right? Thank you.
Thank you, Dr. Ye, for the question. So regarding the details for the regulatory consultation service provided by us, the CDE, the more details is about when we receive, like I said before, if you have any questions or doubts in your package or you want to have a discussions, you may submit the application through our website. And once we receive your applications, we will have a pre-meeting with the sponsors to discuss possible issues or topics. And based on those topics, we will decide what kind of technical dossier or data will be required for the review team. And after we and the applicants or sponsors come to an agreement or a consensus about the payment and the data, then when we receive the payment and the data from the sponsors, the formal consultation service is kicked off at day 0. And we will provide a written opinions from CDE for the applicants at day 56. And we will also arrange a meeting with sponsors to discuss further about the written opinions in the day 60. And the sponsors also can raise questions about the written opinions they receive from CDE to discuss further in the meetings. And we'll also provide the meeting minutes in the day 70 or within the 10 days of the meetings, because not all meetings can have at the day 60. So I want to add a bit more about the charging fees. So the fees will be depending on the sections involved, like if applicants only have the clinical sections, so the price would be one kind, but if like clinical section and PD, PK sections is involved, then the price would be higher. So that's some details about the regulatory service we can provide. Thank you.
Okay, thank you, Ji-Ren. And then I think we can continue to the question from the online audience.
Okay, so the second question from our online participant is from Sharon, RA from CMIC, and this question is addressed to Dr. Jinxun Chen. The question is, under Rare Severe Disease Priority Review Voucher Program, it is not necessary to submit BSE application in advance. However, the ethnic difference will be evaluated during NDA. So there are two questions. The first one is, does that mean sponsor still has to submit BSE report followed BSE checklist during NDA? And the second question is, as the last mentioned question, indications are granted rare severe disease priority review. It may be possible that there is no Asian population in clinical data pool. Can the sponsor get approval letter under such situation? Any suggestions?
Okay. The first question is that whether BSE submission is required for rare severe disease product. Okay. According to the regulation, the sponsor need not to submit bridging study evaluation, but the ethnic difference will be reviewed in NDA. So when you prepare NDA dossier, you must include all this material required for bridging study evaluation in the NDA submission. That's my answer to the first question. But of course, the 12 item checklist, bridging data package, and your self-evaluation reports. Okay. The second question is, I think you want to ask whether the product can be approved without or with only very few East Asian subjects. We have flexibility for orphan drug. And this principle will be also applied for such cases. And maybe the number of the number of subjects of East Asian could be flexible, and we will consider the disease itself, the dose titrate, the apparent the difference of PK. But for some disease, if the drug is not very toxic, then I think you can use health volunteer to conduct traditional PK study. So for severe rare disease, you may conduct a 12 subjects traditional PK study in healthy volunteer without using patients as a subject, as a source of subjects. So that I think basically the clinical review will review the PK comparison first. So if you can convince the regulator that there is no no meaningful difference from a pharmacokinetic point of view, or if you have pharmacodynamic data, that will be better. Okay, so I would like to say again, if you have such program, please consult CDE earlier.
For this question, Ms. Wang, do you want to add some comments?
And I want to echo this question. As Dr. Chen mentioned, healthy PK might be one of the evidence. And I want to mention one situation. Sometimes drugs with a rare disease indication, they probably have another proposed indication is normal and not a rare disease. Might be there will be Asian data, could be a bridging data. So if you could provide a whole package about the two indications, maybe the bridging data in the other indication, which is not a rare disease, could be a supportive information. And if the data, Asian data was sufficient to clarify the arsenic sensitivity, and the PK behavior was similar between the two indications, or might be the result could be extrapolated to the indication that you propose, which is rare disease. So I have seen this kind of a situation in BSE. So indication extrapolation might be one of a possible solution if you have another proposed indication and for development in Asian area is one of the solution. Thanks.
Okay, Dr.
Let me make a very quick comment. On a rare, severe disease, it doesn't sound like a normal disease. It means probably most regulatory apply doesn't fit. The best strategy, you make a friend with the TFDA and CDE. You have to work together. That's the way I see. Because of the rare, severe, its meaning is not common.
I would like to additional comment. And since the enrollment of the rare disease is very difficult, local in Taiwan, so sometimes for a medical need, we will waive the bridging study under certain condition. For example, we may require post-marketing data of its agent. And I noticed that there is a trend to use the data from compassionate use program. The compassionate use program the purpose of compassionate use program is not for drug development, but usually this is for rare disease. And the data could be obtained from the hospital, and is a real world data. So if the compassionate use program is well designed, and if the prescriber can make sufficient data for the patient, that's real world data may be very useful as a supplement for post-marketing requirement if the Eastern Asian data is not sufficient during the BSc submission.
Thank you for another very important comments. And I think we can go further to question 3.
Okay, the third question is also from Sharon, RA from CMIC. And the question is addressed to Ms. Wang. And the question is, in your lecture, you mentioned sufficient Asian population should be included in clinical study. And does it mean sufficient Asian population in the pivotal study, or it can include all Asian data, including phase 1 and phase 2 study? And also, the second question is that, in your experience, can you provide an estimated percentage range which would be sufficient?
Thanks. And for the answer to question 1, the sufficient Asian population is included in all development phase. From PK perspective, if you can provide a comparison with intensive PK sampling and a PD comparison in target patient in early phase, maybe phase 1 or phase 2, and those data could clarify the difference between races, and we won't asking for more data in phase 3. And the second question, in my experience, and you want to know an estimated percentage range of Asian will be sufficient. As much as possible, normally we expect to see more than 10% of East Asian in population PK analysis. 10% in population PK analysis. However, for some rare disease with low incidence, we understand that the Asian population data was limited, so please have a consultation with CDE. Thanks.
Okay, thank you, Ms. Wang. We provide the general principle and for your consideration. And I don't know if for the online question, if our answer is sufficient or not. Please also, you can write down on the online. And for this question, Dr. Jian would like to comment further, please.
From my talk, I think I emphasized several times on the markets in Japan. I understand the BSE, the strategies also start with the PK, the drug exposure, be specific. So I'm wondering, is that possible maybe for CDE or for TFDA to investigate the central PK profile between Japanese and Taiwanese? I think that information will be very helpful for the whole biotech Taiwan to have some basis to move their drug forward. I think the important thing is indicating the two countries, the response to the drugs, not that much difference. That I will see the fundamental for BSE apply to the future approval in multiple countries. Bye-bye.
Okay. From clinical point of view, we in bridging study evaluation, we usually recommend the sponsor to pool the data of East Asian. But when we make assessment, make for evaluation of the ethnic difference, there are two major components. One is the ethnic difference of efficacy. The other one is for safety. So if you want to pool the data from phase 1 to phase 3, you must make sure that the target dose should be the same for the clinical trial you used. Usually for phase 1 study, the target dose may be included in the phase 1 study, but the subject number is not enough, and the time of exposure may be not long enough to show the efficacy. Most important issue is that efficacy cannot obtain from healthy volunteer. And even if you use patient as a phase 1 study subjects, you need appropriate endpoint as used in the confirmatory trial. So that's the consideration when you pulling the data from phase 1 to phase 3. And for safety, that will be more flexible because you can adjust the exposure time. But again, the early phase in the early phase trial, you may have enough exposure time for certain dose. So maybe some adverse event. So you cannot maybe some adverse event were not yet observed in the short-term clinical trial. Okay. But for safety, the pooling of data will be more flexible. Okay.
Okay, very good comments. And how about we go to next question?
Okay, so the next question is from Julia Chu, our AM manager from GSK, and this question is addressed to Dr. Jixun Chen regarding clinical consideration. The question is there any specific consideration in the review of vaccines BSE? Is Taiwanese data mandatory, or the applicant can fully rely on East Asian data without Taiwan data in vaccines BSE to demonstrate no ethnic difference? Thank you.
Sorry, I have little experience for vaccine, so maybe Dr. Jan will answer this question.
Okay, the question from GSK is about specific to vaccine BSE consideration. In general, I would say there's no PK concern from review perspective. That's the first point. And in terms of ethnic factors consideration, there's less intrinsic factor concern based on our review experience. So the critical issue goes to the extrinsic factor, especially when we are talking about the infectious disease. The epidemiology is so critical. It could be region-based. It could be very different from different countries. And the socioeconomic status always probably played a role, and the society immunity. So in principle, again, the Taiwanese data is from regulation is not mandatory, but it's highly valuable. So if the sponsor doesn't want to conduct any study in Taiwan, but you have to address the issue what I just mentioned. You have to address the issue, the Taiwanese data. There's no Taiwanese data, but we could bridge the gap using the other East Asian data. That's the point. Okay, thank you. That's my comment.
I will add some comments for this question. Like Dr. Zhang said, and I suggest you more consider the prevalent strain across in a different country. So it's most related to the extrinsic factor, like Dr. Zhang said.
Okay, thank you, Dr. Jen, Dr. Xu. I think this afternoon's talk not only is very comprehensive, our question this afternoon is also very comprehensive, from rare disease, severe drug for severe disease, and vaccine. I think those are more specific category for the BSE consideration. I think probably that's for the online questions. Yeah, I don't know if you have further question from the floor.
If any further question? Yeah, if there is no further question, in the end, I will invite all the speakers to give us some comments. And first, I will invite our guest speakers, Ms. Watt. Please.
I would like to share my sincere appreciation to the organizing committee for this opportunity to share the perspectives from the industry over the past 20 years implementation in Taiwan. I think looking ahead, there's a lot of opportunity for the industry and also the Taiwan FDA and also the other regulatory authorities in Asian region to collaborate to bring trials to Asia to understand better the disease patterns in Asia, to understand the influence of ethnic factors in drug design. But I think if we can have more platforms to create sharing of experiences so that we can understand each other's issues and challenges, and think about the collaboration opportunities ahead. Also looking at the opportunity of ICH 17 can bring to the design of multi-regional clinical studies. I think this would be really a big opportunity. So I look forward to more opportunities in the future for this kind of platform. Thank you.
Thank Ms. Watt. And Dr. Jian, please.
First of all, I want to thank you for the committee. Very interesting conference. I want to comment a few things. 10 years later, if we have a 30 years the ceremony of a BSc, I will imagine this is totally different. Perhaps to give a little bit background about the clinical development. The preclinical IND submission in phase 1, 2, 3, that was established about 80 years, 1983 or 84. When Roger Williams, I think somebody, a speaker mentioned this morning, when he was director of the FDA, and also Janet Woodcock was director of CDER. At that time, very focused on the GMP and GCP, GLP at that time. So there was established phase 1, 2, 3. But nowadays, 30 years later, the technology advances so fast, including generic profiling, other genomic thing. The thinking of the global drug development is no longer 1, 2, 3. Perhaps 1, 2 combined, or 1, 2, 3 combined, something like that. So when the time to apply for BSE will be very interesting, I would say. You probably will see the very few subjects from Asian and Caucasian in the very early stage. That's what we're doing right now in the phase 1 study. I think that speed will faster and faster. Even the global big pharma, even smaller, more flexible biotech. That's what we are thinking. So nobody can wait for 10 years to get product approval. Everybody's shooting for 3, 4 years. So you can imagine that very fast. So I think 10 years later, we probably still here, but the whole white hairs. I will imagine the BSc ceremony will be totally different. We probably will talk more advanced technology and application to the global drug development. That's what I see.
Yeah, thanks, Dr. Jian. And how about my colleague, Mr. Manager Yang Jiren?
Thank you, Dr. Xu. So again, I want to emphasize that I'm so honored to be given this opportunity to be here as a speaker. And again, as a newbie here, I really learned a lot from all of the speakers across the industry, the academic, and also our senior colleagues. So I learned a lot today, and I'm eager to learn more. So thank you.
And Ms. Wang?
And I'm glad to have this opportunity to share experience, and I did learn a lot in this conference. And I want to share one experience in my review experience. And sponsors like to cook data in many ways in order to reducing the PK difference between races. And the most way laid down and show in the in bridging data package is to body weight normalize the PK data. But however, for a drug which was fixed dose given as a fixed dose, a body weight normalized PK data was only a supportive, because the body weight of Asian population usually is lower than that of Caucasian. So body weight normalized PK data is not that meaningful from PK perspective. It's only a supportive. So I want to share this experience to sponsors that you have to depends on your indication and dosage regimen and choose the proper way to cooking your data. And that's why I mentioned in the speech and other speakers' speech that how to prepare proper bridging data will, and also involve Taiwan in your bridging data. Let's get a higher waiver rate in Taiwan. Thanks.
Dr. Chen.
I have a few comments. First, bridging study evaluation is reviewed case by case, considering various factors as I mentioned in my presentation. Second, traditional PK data are essential and will reviewed before clinical comparison. If there is critical difference from PK data or PD data, the target dose might be changed. Third, the amount of East Asian data varies case by case. So I cannot tell you the one magic number, how many East Asian patients needed. MRCT is encouraged, but you have enough subject for subgroup analysis. So you may pre-specify the region you want to pool. For example, you may pre-specify Taiwan, Korea, and Japan, the 3 countries, for pooling for East Asian subgroup analysis. And I strongly suggest that if you pre-specify the region, then you have to stratify it by the region when you enrolling the subject. In conclusion, early consultation to CDE is highly recommended. As early as possible, even during phase 1, you may consult CDE. Okay, at least we will give you opinion about how to design the PK study. Okay, thank you.
Yeah, okay. Any further question? Okay. Finally, I will thank all the speakers today again, and thank all the audience in the meeting room and online. And thank you so much.
Thank you. That concludes the panel discussion. Please give a big hand to all our panelists and our moderators. Thank you very much. Thank you. Please return to your seat. Thank you. 非常感谢综合座谈的主持人以及所有的与谈人，非常感谢为我们带来精彩的对话，也非常感谢线上还有现场朋友非常热烈的提问。So the conference today is coming to an end. Thank you so much for your generous participation. And once again, we thank our distinguished guests from Japan, Singapore, and Taiwan, and all the attendees on site and online to join us and mark the 20th anniversary of Bridging Study Evaluation. Thank you so much. And please remember to take your personal belongings when you leave the conference room. We wish you a lovely day. Thank you.